Hi kids. Hi everyone, are we live? Hi kids, are we live? Hello. Guys, can you hear me? Hi everyone. A very good evening to all of you. Can you see me? Can you hear me clearly? Yes? Good evening, good evening, good evening. Welcome to Unacademy NEET English. I'm your biology educator Ambika Sharma, and today we are going to complete biological classification. We will talk about the Protista, fungi, viruses, lichens, viroids, prions. So this is what we are going to discuss today. So first of all, tell me everyone, how are you? How was your Sunday? I think I have given you the enough time to enjoy your Sunday. Isn't it? Isn't it? You got enough time to enjoy your Sunday. So that is why I kept the class in the evening, that in the morning you can get up late, right? You can utilize your day in other activities, and in the evening let's revise sometime. For some time, let's revise the things, isn't it? Okay, so let me see your chats. Okay. Good evening. My day was good. I was just waiting for your class. So tell me guys, how are you feeling? We have completed one chapter. This is the second chapter that we are going to finish, and after that we will start the plant kingdom, animal kingdom. So basically we are about to finish our first unit, that is diversity in the living world, and there will be a test for that unit also. There will be a test for that unit also, right? So whatever we are going to complete here on this channel, we are going to solve the questions from that too. It's good, isn't it? Isn't it? Okay, so tell me guys, you have revised the chapter, right? So I'm going to ask you ask few questions. So my first question is, who tried to classify the organism for the first time in a scientific manner, basically? Yes, in a scientific manner, basically, who tried to classify the organisms for the first time? And we have so many students and number of likes are quite less, so everyone just hit the like button quickly, and if you are new to our channel, do subscribe our channel, and let's finish this chapter today, bache. And please keep your notebook with you, your NCERT with you, right? We are going to mark all the important points, and let me tell you, here in this lecture, right, we are going to follow the old NCERT, not the rationalized content. Right, we are going to follow the old NCERT, and what is the reason behind it? Because NTA did not announce anything related to syllabus reduction yet, right? So that is why what we have to do, we have to read the NCERT, we have to complete the NCERT. Bache, you will get the PDF once I will finish this chapter. You all will get the PDF. So first of all, answer my question, everyone. Right, everyone just answer my question, that who tried to classify the organisms in a scientific manner for the first time? Yes, Aristotle. Very good, very good. Okay, my second question is, who gave the four kingdom classification system? That's my second question. Who gave the four kingdom classification system? The word is four kingdom. And guys, speed up, all of you just answer the question. Answer the question and hit the like button. Copland, very good. So, who gave the five kingdom classification system? Who gave the five kingdom classification system? Yes, everyone? Who gave the five kingdom classification system? Thank you so much, bache. Thank you so much. R.H. Whittaker, very good. So let's start the lecture, bache. So first of all, what you need to know, what we have covered in the last class, let's revise that thing, right, bache? See, we have started biological classification, isn't it? The name of the chapter is biological classification, and here in this chapter we talk about the classification. We start from Aristotle. Right, we start from Aristotle. He He was the first person to classify the organisms in a scientific manner, although it was not a proper system. On the basis of, you know, when it comes to plants, he just checked the morphological characters. Right? When it comes to plants, he just checked the morphological characters. Right, bache? So in the case of plants, as per Aristotle, right, he divided them into 3 different groups on the basis of their size, like herbs, right, shrubs, and the trees. And when it comes to the animals, so on the basis of red blood, right? So one is anima and another word is what? Enima. This is what we have covered in the last class, isn't it? This is what we have covered in the last class. So when it is A, the word is A means without. So anima means organisms without red blood, without RBCs. Is that clear? Without RBCs. Now when you talk about the enima, so here we are going to consider the animals which are having the red blood. Okay, which are having the red blood. So now you know that these systems, like this system basically, particularly the Aristotle system, we cannot accept it. Because we have diversity of organisms, variety of organisms, right? Organisms are so diverse. We cannot categorize them on the basis of their morphological characters only, right? Right? We have to look for the anatomical characters, the phylogenetic characters, and there are many other things also that we need to check. So obviously, it is not making sense, isn't it? It is not making sense, isn't it? Isn't it? Isn't it? So after Aristotle, what was there? 2 kingdom classification system, then 3 kingdom classification system, then 4 kingdom classification system, and finally there will be the 5 kingdom classification system, and there is one more, that is 6 kingdom classification system. Right, bache? This is what we have. Okay, 2 kingdom, 3 kingdom, 4 kingdom, 5 kingdom, 6 kingdom. Isn't it? So when it is 2 kingdom classification system, so you know that Carl Linnaeus, isn't it? Who gave it? Carl Linnaeus. But the scientist's name is important. So please revise it with me. Revise it with me. 2 kingdom means Plantae and Animalia. 2 kingdoms means Plantae and Animalia. Next is 3 kingdom. It was given by Ernst Haeckel. Okay, it was given by Ernst Haeckel. Right? 3 kingdom classification system. He introduced kingdom Protista. And today we are going to talk about the kingdom Protista. Then next is 4 kingdom classification system given by Copland, and he introduced what? He introduced kingdom Monera. And then comes the 5 kingdom classification system given by R.H. Whitaker. Right, R.H. Whitaker, he introduced the kingdom Fungi. And when it comes to the 6 kingdom classification system, then the scientist's name is Carl Woese, and he basically divided the bacteria. Right, he divided the bacteria into two different kingdoms. Right, he checked the bacteria as per him. One was Archaebacteria, one was Eubacteria. So he divided that bacteria. Right, he divided that bacteria. And when it comes to the 6 kingdom classification system, he also introduced the highest category as domain. Right, highest category as domain. So 3 domains were introduced by him. Okay, 3 domains were introduced by him. Okay, so this is for the quick revision, bache. This is for the quick revision, and as I said, it is important. Okay, it is important and you need to remember it. Right, so let's have the quick revision. So here you guys can see this is what we are going to discuss in this particular chapter. Right, so 2 kingdom, 3 kingdom, 4 kingdom, 5 kingdom, and these are the exceptions. Okay, these are the exceptions to 5 kingdom classification system. Your viruses, viroids, prions, lichens, they are not the part of 5 kingdom classification system. They are exempted from 5 kingdom classification system. Then kingdom Monera, that we have completed in first part. Right, and today we are going to talk about this and this. Okay, so today we will be starting the kingdom Protista, and after that kingdom Fungi. Okay, so this is what we need to start today. So Srinath is asking, ma'am, what are those 3 domains? 3 domains, they are very easy. Remember? Anyone in the class? We have discussed that in the last class. Can you answer this? Very good. PG, Vijay. Very good. One is archae, another one is bacteria, and third one is eukarya. Archae, bacteria, and eukarya. These are the 3 domains. Okay, these are the 3 domains. Is that clear? So here you people can see, right, because today we will finish the chapter, so definitely after this class you will get the PDF of these 2 lectures. Fine, for your quick revision. So these tables are given, bache, and while making your notes, please include the points, right, all the important points that I used to mention. Moreover, try to make your notes in this way. Try to make the tables there, flow charts there, write some mind maps there, so that you can save your time and you can revise it quickly. Is that clear? Fine. Is that clear? So now here we go, and let's start the Kingdom Protista, but have a quick revision of this part. Right? You know that in our syllabus, what is there? 5 kingdom classification system. Isn't it? What is there in our syllabus, bache? 5 kingdom classification system. This is what we discuss in our syllabus. Right? So here you guys can see Kingdom Monera, Protista, Fungi, Plantae and Animalia. Right? Kingdom Monera, Protista, Fungi, Plantae and Animalia. This is what we have discussed, right, in the last class. So if you are new to this class, do check the last session, first thing. Secondly, even if you are checking the recorded session, please try to revise both the parts all together. Okay? Okay, and ma'am, please take the Menti. Bache, see, honestly speaking, I am not in the favor of Menti. We are going to like, I don't want the Menti sessions to be there very frequently. The reason is, firstly, we have to finish the theory. We have sufficient time to complete the theory, and later on we can keep it. But yes, after finishing this particular unit, I will ask you the questions, right? There will be prizes and all, right? Then I'll keep it in a Menti format. Okay? Okay, now please focus here, everyone. Just focus here. So Monera, Protista, Fungi, Plantae, Animalia. So now answer my question. How many kingdoms are having prokaryotic cell? Quick. How many kingdoms are having prokaryotic cell? And guys, be very quick. Yes. And do hit the like button, do subscribe our channel and share this video link with your friends as well. How many kingdoms are having prokaryotic cell? Yes. Quick. Yes, everyone. How many kingdoms are having prokaryotic cell? I can see many students are beautifully giving incorrect answer. Very good, bachcho. Very good. It is such a simple question and you are giving incorrect answer. Very good. I'm so proud of you. Keep it up, huh? It is just one kingdom, and that is Kingdom Monera. So, bache, it is only Kingdom Monera which will contain prokaryotic cell, right? And when it comes to the prokaryotic cells, bacteria is the sole member of Kingdom Monera, right? Bacteria is the sole member of Kingdom Monera. And in the last class, everything related to bacteria, like the structure of bacteria in detail, right? Different types of bacteria, archaebacteria, eubacteria, which will include your actinomycetes, ray fungi, which includes your cyanobacteria, photosynthetic bacteria, which includes your, yes, what else? Mycoplasma. We have discussed, we have covered everything, right? So today let's start from this particular portion. Let's start from Kingdom Protista. Okay, let's start from Kingdom Protista. So first of all, just repeat it with me. CDESP. All of you, just repeat it with me. CDESP. CDESP. All of you, just type it in the chat section and be very quick. Type it in one go. CDESP. 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 Right? And in this flow, you have to remember it. Right? So, whenever we talk about the Kingdom Protista, what do we have? We have CDESP. What do we have? We have CDESP. CDESP. So, what is the CDESP? C stands for chrysophytes. D stands for dinoflagellates. E stands for euglenoids. S stands for slime molds. P stands for protozoa, right? P stands for protozoa. So in Kingdom Protista, what are we going to study? We are going to study CDESP. What is this CDESP? Chrysophyte, dinoflagellate, euglenoid, slime mold, protozoa. chrysophyte, dinoflagellate, euglenoid, slime mold, protozoa. So this is what we are going to discuss in the kingdom Protista, right bache, in the kingdom Protista. Now here you guys can see the beautiful images, right? These are the representative organisms here and we will discuss them in detail. Okay, so let's start the kingdom Protista. So first of all, when it comes to the kingdom Protista, so it also includes unicellular organism, right? It also includes unicellular organisms. See kids, kingdom Monera is also having unicellular organisms. Kingdom Protista is also having unicellular organisms. But the only difference is in the kingdom Monera, bacterial cells are there, prokaryotic cells are there, and when you talk about the kingdom Protista, you will be having the eukaryotic cell, which is the advanced cell, right? And in other phylums, fungi, algae, plantae, animalia, you will see multicellular organisms, and yes, there are few exceptions. Okay, there are few exceptions. So what are we going to discuss? We are going to talk about the kingdom Protista, right? And it includes what? It includes the unicellular eukaryotes. So eukaryotic organisms will be there, and what are they? What are they? Unicellular. So basically here what can we say? In kingdom Protista, members are unicellular, right, having nucleus, proper nucleus, or you can say that covered with, okay, just write this word only. They are having proper nucleus, organelles, ATS ribosome in cytoplasm, isn't it? ATS ribosome in cytoplasm, because it is a eukaryotic cell, there will be the proper nucleus, all other cell organelles will be there, and the ribosome in the cytoplasm will be ATS, right? Ribosome in the cytoplasm will be ATS. Right now I am talking about the cell structure here, and moreover, if you talk about the flagella, because we will see the motile organisms also, right? We are not just going to see the non-motile organisms here, we will see the motile organisms here also. So when it comes to the flagella, flagella is made up of one protein, and that is tubulin, right? Here flagella is made up of what? It is made up of tubulin protein. So it simply means that here the flagella is having the microtubules. So if it is having the microtubules, so the arrangement is going to be 9 plus 2, right? Which arrangement will be there? 9 plus 2 arrangement will be there. So what are we discussing? Kingdom Protista, and right now we are talking about the cell structure here, right? We are talking about the cell structure here. So proper eukaryotic cell, as I said, nucleus, organelle, ATS ribosome. When it comes to the flagella, right, flagella will be what? It is made up of tubulin protein, it is made up of microtubules, 9 plus 2 arrangement will be there. So just compare it with kingdom Monera, right? Just compare it with kingdom Monera. In the kingdom Monera, we have what? We have the, yes, we have the flagella, but that flagella is made up of flagellin only, right? That flagella is made up of flagellin only, right bache? So now if I talk about the habitat, so mostly they are what? Mostly aquatic, isn't it? Mostly aquatic, isn't it? When you talk about the kingdom Protista, the members of kingdom Protista, what are they? They are mostly aquatic, and moreover bache, here in this kingdom, right, in this kingdom, you will study the members just like plants, just like animals, just like fungi, right? It is even given in NCERT. When you talk about the kingdom Protista, you will study different types of unicellular organisms there, and you will see the similarity with the animals, you will see the similarity with the plants, even you will see the similarity with the fungi. So here even we used to say, when it comes to kingdom Protista, it is going to show us all the characters of all other kingdoms, right? Characters of all other kingdoms. So basically this phylum is having no boundaries, Okay, bache Harsha. So this phylum is basically having no boundaries. Is that clear? So as I said, what are they? They are mostly aquatic, right, bache? Mostly aquatic. Flagella is there with 9+2 arrangement, right, bache? Flagella is there with 9+2 arrangement. Even they can have, right, cilia also, pseudopodia also. We will discuss the different examples. As of now, I just highlighted this part only. And in this kingdom, organisms are like plants, fungi and animalia or animals, right? You will see the similarities. So I am just writing it down in short, right? So this kingdom is having no boundaries. It is important to know. This kingdom is having no boundaries. Is that clear? Yes, everyone. Is that clear? This kingdom is having no boundaries. Clear, bache? Clear, bache? What is 9+2 arrangement? Bache, 9+2 arrangement here is of flagella, right? So basically here flagella is made up of tubulin protein. So tubulin will polymerize, it will form microtubules. That microtubules will in return form flagella. So you will see that on periphery there will be two microtubule. Basically there will be the doublet, and in the center there will be the singlet. Basically 2 single microtubules will be there. So if you want to know about it, right, I'll start the cell chapter. In the cell chapter we will discuss it in detail. Fine, bache, we will discuss it in detail. Okay, okay ma'am, no boundaries. No boundary means here in this particular kingdom the organisms, they are so diverse, right? So you will see different type of organisms here in this kingdom Protista, right? That is why we use this word, that they do not have the proper boundaries. Okay, okay, it just act like a link in between animals, fungi and the plants. Okay, it just act like a link in between the animal, the point here. That is the point here. And moreover, when you talk about the Protista, you will see, you know that organisms are what? They are mostly aquatic, they are small sized, they are aquatic, and they will passively flow over water surface, right? What will they do? They will passively flow, right? Do you know the meaning of this particular statement, passively flow? Like let's say there is a water body, right? There is a water body. Now these organisms, just with the water current, they will move freely, they will move. And for such organisms we use the word plankton. Are you aware of this word, plankton? Like we know about the phytoplankton, zooplankton. So here what is the point? Basically you will see free floating organisms, and for free floating organisms we use the word plankton, right? We use the word plankton. So they passively flow over water current. So that is why we use the word plankton. So in short I am writing it, so you guys can just note down the complete statement. Fine? Fine. See bache, the word is plankton, right? So if it is performing the photosynthesis, means it will be having plant like character. You can use the word phytoplankton. But if it is holozoic, if it is taking solid food, means animal like character, then you guys can use the word zooplankton, right? Then you people can use the word zooplankton. So these are some general characters of the members of the kingdom Protista, which includes unicellular eukaryotic organisms. Ultimately this is the point. Now the next part is their nutrition, right? That what type of nutrition do they have? One student was asking na that, ma'am, I didn't get the meaning of the phylum with no, the kingdom with no boundaries. I'll tell you now. Now let's focus on their nutrition. So when you talk about their nutrition, so you will see autotrophic also, you will see heterotrophic organisms also. Are you getting it? You will see autotrophic organisms also, you will see the heterotrophic organisms also, right? So basically we divide them into groups when it comes to the, right, when it comes to the what? When it comes to their nutrition. Like firstly, the broad category, the two broad categories are photosynthetic, right? First is photosynthetic Protista. First is photosynthetic Protista. But can you tell me which Protista are photosynthetic? Which protists are photosynthetic? Yes, bache, they even have the cell wall, but not all the organisms are having the cell wall. We will talk about the individual characters as well. But first of all, tell me, can you tell me the name of the organisms which are photosynthetic? Right here, which protista are photosynthetic protista? This is what I'm asking. So please kindly answer it, bache. Kindly answer it. Anyone? Yes? Tell me. One is photosynthetic protista. So in photosynthetic protista, what do we have? We have three names. Chrysophytes. I told you na, CDESP. So chrysophyte, D stands for dinoflagellates. D stands for dinoflagellates, and when it comes to CDE, E stands for euglenoids, right? E stands for euglenoids. So they are basically phototrophic. They are basically autotrophic. They are going to make their own food. They are going to make their own food, photosynthetic protista. And here, when it comes to the euglenoid, you will see mixotropic nutrition. I will explain each and every term to you, don't worry. Right, here you will see mixotropic nutrition. And now the another category is of ingestive, right? Another category is of ingestive protista. They will eat up the solid food. So you will see the protozoa there, right? What will you see? The protozoa there, right? So among protozoans, we will be having the ciliates, we will be having the flagellates, we will be having the sporozoans. So this is what we will see here. So one category is photosynthetic protista, bache, right? Another category is ingestive protista, where your protozoans are also, protozoa is also included, right? Where your protozoa is also included. And when it comes to the slime mold, right? When it comes to the slime mold, so bache, the slime mold is saprophytic, right? Saprophytic. Is that clear? When it comes to the slime mold is what? Slime mold will be saprophytic. Is that clear? Yes, everyone. Is that clear? If there is any doubt here, do let me know. Yes, bache, do let me know. So it will be saprophytic. Okay, saprophytic. Quick. Right, saprophytic, right, which will feed on dead and decaying organic matter, right? So when it comes to the slime mold, they will be saprophytic, or you can also use the word saprobic, or even you can say some are parasitic also. Not just the slime molds, they are parasitic. There are some protozoans also. We will discuss that examples. Okay, so that type of nutrition is there. So on the basis of nutrition, we also divided. And here, bache, right, for in the case of euglenoids, I told you that you will even see the mixotropic nutrition. So understand here, photosynthetic protista, which are going to perform the photosynthesis. So we also call them as holophytes, right? We also use the word holophyte for it. When it comes to the photosynthetic protista, we also use the word holophyte for it. Holophyte nutrition means plant-like nutrition where they are performing the photosynthesis. Now when it is ingestive protista, basically they are going to inject the solid food. So holozoic will be the word here. Holozoic nutrition will be the word here. So here you will include protozoans, right? Protozoans. And when it comes to the slime mold, obviously slime mold, it will feed on dead and decaying organic matter. Right, and I am not saying that protozoans are not parasitic. They are. So one by one we will discuss the example. As of now, broadly, this is how we are categorizing them. Are you getting it? This is how we are categorizing them. Right. So these are some general characters. Is that clear? Is that clear? Now the next part is their life cycle. Next part is what? Next part is their life cycle. But do you know about the life cycle of protozoans? Do you know about the life cycle of protozoan? Yes? Anyone? Do you know about the life cycle of protozoan? It's very simple. In the case of life cycle, there will be two words majorly. Like one is zygotic meiosis. Zygotic meiosis, but these are basic things and I think you all should know about it. And one is gametic and one is what? One is gametic meiosis. Do you know about it? Yes. Do you know about it? One is your zygotic meiosis, another is your gametic meiosis. See, if you don't know anything related to meiosis, on this channel initially when we were catering the NEET 2023 students, at that time I have taken one session where I have explained the mitosis meiosis in detail. So you all can check that particular session if you want clarity on the term meiosis, right? And later on, no doubt, we will soon start this chapter also. Fine, bache. So life cycle is zygotic meiosis, gametic meiosis. On the basis of these two things, we have different type of life cycle. Like if it is the zygotic meiosis, then the life cycle will be haplontic life cycle. I know you must be thinking that, ma'am, these terms are in the case of plant kingdom. No, these terms are in general, these terms are some general terms, and I think you all should know the meaning. Now when it is the gametic meiosis, then what is the word? Then there will be the diplontic life cycle. Right, what will be there? There will be the diplontic life cycle, and when it is the haplontic life cycle, you will see the example of dinoflagellates, and when it is the gametic meiosis, you will even see the example of diatoms. Now I will explain you this. See, it's very simple. The word here is zygotic meiosis. Just focus on these two words here. Actually, see, Protista. Protista is the first kingdom. Protista is the first kingdom where you will see sexual reproduction. Do you know that? It is the first kingdom where sexual reproduction is seen, right? Proper sexual reproduction. Now some students must be thinking that, ma'am, in the kingdom monera also under the title sexual reproduction, we have discussed conjugation, transformation, transduction. But basically, in the case of kingdom monera, where we talk about the bacteria, but that bacteria, that bacteria, they do not form gametes. There is no proper sexual reproduction. No doubt a type of genetic recombination is there, right? No doubt a type of genetic recombination is there. So that is why we used to say the sexual reproduction is there. But actually the proper sexual reproduction you will see in the kingdom Protista, right? Here we will see the gamete formation, gametes will fuse, right? There is the syngamy, zygote formation will be there. This is what we are going to see. Okay, so now the word here is zygotic meiosis. What is the word that we have here? The word here is zygotic. The word here is zygotic meiosis. Now, see, you know that what is meiosis? Meiosis is the reductional division, isn't it? What is meiosis? Meiosis is the reductional division. What is the meaning of reductional division? It will reduce chromosome number to half. What will it do? It will reduce chromosome number to half. Isn't it, bache? Do you know about it? What will it do? It will reduce chromosome number to half. Chromosome number to half. This is the meaning of meiosis. Now, let's say if any organism is having mean body as haploid. This is what we even discuss in the case of plants, and in next chapter in kingdom Plantae also we will discuss it, but try to understand the concept here, right? Try to understand the concept here. You know that there are some organisms and in their case the main body is haploid. Like take our example, we are human beings, right? In our case, if you take out any cell from our body other than gametes, if you take out any cell from our body, it will be diploid. It will be having two sets of chromosome, one from mother, another one from father, one from mother, another one from father. This is what we have. Yes, this is what we have. So what are we? We are the diploid organisms, right? We are the diploid organisms, but we have the example of certain organisms where they are a haploid. Right now we are dealing with kingdom Protista. So in kingdom Protista, the members of kingdom Protista, they are unicellular, right? The members of kingdom Protista, they are unicellular. So in their case, in kingdom Protista, that organism can be haploid, it can be diploid. You cannot just justify this, right? You cannot just say that all the organisms will be haploid or all the organisms will be diploid. No, you have to focus here that in some organisms zygotic meiosis is there, gametic meiosis is there. Means basically the organisms that belongs to kingdom Protista, in their case their life cycle is different. It is not all same, it is different. What you didn't understand, Madhava? Right, it is not different. So now just focus here. So I am saying organism is haploid. So let's say if any organism is haploid. Now this is a haploid cell. It needs to form the gamete, right? Even you can take any example. You can take the example of a multicellular organism. You can take example of a unicellular organism. Whatever example you want to take, you guys can. So just focus here. What is the point here, bache? Now this is the haploid organism. What is it? It is the haploid organism, right? Now this haploid organism, it needs to form the gametes. How will it form? How will it form gamete? How? Can you tell me? Can you tell me? It is a haploid cell. It is a haploid organism. This organism is going to form the gametes, right? This organism is going to form the gametes. How will it form the gametes? Very good, Priyanka. Excellent, bache Priyanka. Very good. So obviously it is already haploid. So if it needs to form the gametes, it will form gametes by mitosis. It will form gamete by mitosis. It is not going to use the meiosis then. It will use mitosis. So after mitosis, what is going to happen? It will form the haploid gametes. So what will happen? These haploid gametes, because you know that gametes are always haploid in nature. Gametes are always haploid in nature, right? Whatever is the chromosome number of the parent cell, the gamete will have haploid number of chromosome. But here already we have a haploid organism. So by mitosis it will form the gamete, and then what will happen, bache? These gametes will fuse together. They will form zygote. You know that it is syngamy. What is it? It is syngamy. What is it, bache Priyanka? It is syngamy. So now this zygote is diploid. It is formed by the fusion of two gametes. So obviously what is it? It is diploid. It is diploid. So now this zygote will immediately undergo meiosis. This zygote, it will immediately undergo meiosis. What will happen? This zygote, it will immediately undergo meiosis. So that is why we are saying it is the zygotic meiosis here, right? That is why we are saying it is the zygotic meiosis here, because zygote is undergoing meiosis. Now you tell me, if zygote is undergoing meiosis, we can also use zygospore word for it. If a zygote, you guys can take a note of it. If a zygote is undergoing meiosis, we can also call it as a zygospore. It is not zoospore, it is zygospore, right? Specified. It is a zygospore. Okay, it is a zygospore. So this zygote will undergo meiosis. So if it is undergoing meiosis, what will it form? Yes, bacho, what will it form? Everyone, Telugu guy reaction. Bache, already I have explained in the case of Monera. They are not going to use the mitosis. Seriously, killer science, are you serious? Of course. So what will happen? It will form 4 haploid spores. It will form 4 haploid spores. The number will be 4, not 2. It is meiosis. 2 rounds of division are there. So ultimately we will have 4 daughter cells. So 4 haploid spores will be there, right? 4 haploid spores will be there, and that haploid spores will again germinate, right? They will again germinate, and what will they form? They will form that organism, isn't it? So this is basically the haplontic life cycle. Got it? This is basically the haplontic life cycle, where the main organism is haploid. So zygote will undergo meiosis, right bache? So it is zygotic meiosis. It is zygotic meiosis. We need to study it in the case of what? In the case of your plants. we also need to study this. So you better note it down. So it is zygotic meiosis. It is zygotic meiosis. Is that clear? The first case. Now the second case is, diplontic life cycle. The second case is what? It is the diplontic life cycle, just like us. Even we have the diplontic life cycle. Now, what is going to happen here? The main organism, the main cell will be diploid here. Right, bache? Because we are talking about the kingdom Protista, and you know that it is made up of unicellular organism. That is why I am mentioning here the single cell. So, what is going to happen? This diploid organism, with the help of meiosis, now gametic meiosis is there. What is there? Gametic meiosis is there. Gamete will form by a meiosis. So, this cell will undergo meiosis. What will you have? You will have gametes. Isn't it? Isn't it? Sperm, ovum. This is what we discussed. So, the gametes will fuse. That is syngamy. Right? That is syngamy. What will you get then? You will get zygote. What will you get then? You will get zygote. And then that zygote will grow. Okay, that zygote will grow. Fine? Fine, bache? Okay. So this is the diplontic life cycle, or you can say that it is the gametic meiosis. The example is your diatoms, and when it is your zygotic meiosis, so what is the example that you have? You have the example of, yes, you have the example of dinoflagellates. Done, bache? Done, bache? Okay, and here you know that what will happen to this zygote? It's a diploid cell. Here the main cell is also diploid. So this zygote, now it will divide mitotically. Mitosis will be there. It will form diploid cells. They will grow as different individual. Right? They will grow as different individual. Got it? Got it? So gametic meiosis, example is the diatom in the case of what? In the case of your Protista. And here, what is the example? The example is the dinoflagellate. Is that clear, bache? Is that clear? So this is about the sexual reproduction when in the case of Protista. Okay, sexual reproduction in the case of Protista. So next is what? Next is the asexual reproduction. So can you tell me the ways of asexual reproduction? Both the reproductions are there. Firstly, I have explained you the sexual reproduction. Now it is asexual reproduction. So when it is asexual reproduction, the binary fission is very common. Okay, binary fission is very common here. It's very common, and along with that, there will be the sporulation, that is spore formation, and sporulation is in the case of your slime molds. Right here, in the case of your slime molds, you will see sporulation. Binary fission is very common in the case of your diatoms, belongs to chrysophytes, in the case of your dinoflagellates, in the case of your euglenoids, even in the case of your many protozoans, you will find this. Okay, you will find the binary fission. So binary fission is quite common there. Okay, the binary fission is quite common there. Is that clear, bacho? Yes, any doubt? Yes, any doubt? Sure? Are you sure? Sure. Diplontic life cycle. In the diplontic life cycle, diploid cell will form gametes, with the help of meiosis, that gametes will form, zygote will form, right? So zygote will divide mitotically. You will have, daughter cells having a diploid number of chromosomes. So obviously that will grow as an individual. That's all. So this is about the binary fission. So these are some general things related to what? Related to the kingdom Protista. Okay, these are some general things related to what? Related to the kingdom Protista. Now the next thing that we need to understand is, okay, that is chrysophytes. So what are we going to talk? What are we going to discuss now? We will talk about the chrysophyte. This slide is very beautiful, isn't it? Isn't it? This slide is very beautiful. Can you see these different structures here? Can you see these different structures here? We are going to start the chrysophyte. In kingdom Protista, chrysophyte, dinoflagellate, euglenoid, slime mold, protozoa. This is what we have. So we are going to start with the chrysophyte. So see, these are the chrysophytes basically. The different shapes they have, the cylindrical, rounded, right. So this type of shape these diatoms are having. Diatoms are having different type of shapes. So chrysophyte, they include what? They include the diatoms. So here you people can see the chrysophyte. So now let's start it. Okay, chrysophyte. So firstly I will explain you the things and then we will read it from the NCERT, right? Then we will read it from the NCERT. So what are we discussing? We are discussing the chrysophyte. So chrysophyte will include two things. One is diatoms and desmids. What will you study here in the case of chrysophytes? Diatoms and desmids. And desmids are also known as golden algae. Okay, they are known as golden algae. So what will we discuss in the chrysophyte? We will discuss your diatoms and the desmids. What will we discuss? We will discuss your diatoms and the desmids. Isn't it? And I already told you, bache, please keep it in your head, right? Please keep it in your head. They are photosynthetic protistans. Here I have already mentioned you, chrysophyte, dinoflagellate, euglenoids. They are photosynthetic protestan. And here when I was saying mixotropic nutrition, I was taking the example of euglena, right? So euglena is showing both holophytic plus holozoic nutrition, means animal like nutrition also, plant like nutrition also. So it's kind of mix. So we use the word mixotropic, and that is why euglena is considered as the connecting link in between plants and animal. Okay, that is why it is considered as the connecting link in between plants and animals. So what are we discussing? We are discussing the chrysophyte, which will include your diatoms and desmids, and this is the photosynthetic protistans. Okay, so what are they? They are photosynthetic protistans. Is that clear? So diatoms. So what are diatoms? Firstly, let's talk about the diatoms. So obviously it is aquatic. What is it? It is aquatic, right? So when it is aquatic, means it is living in the water habitat. That water can be the fresh water, it can be the sea water, it can be marine, it can be fresh water, isn't it? It can be marine, it can be fresh water. We are basically talking about the diatoms. Okay, we are basically talking about what? We are talking about the diatoms. So it is aquatic, so it can even you can find some diatoms in the fresh water also, in the sea water also, right? In the fresh water also, in the sea water also. But when you talk about the desmids, they will only be there in the fresh water, right? Desmids are only there in the fresh water. Clear, bache? If a water is fresh, clear, you will find the desmids there, right? You will find the desmids there. So right now what are we talking? We are talking about the diatoms. It is aquatic, it can be there in the fresh water, it can be there in the sea water, right, bache? It can be there in the fresh water, it can be there in the sea water. Done? Yes, bache. So in their case, if you talk about the shape, their shape, it varies. When it comes to the shape, it can be circular, it can be trigonal. You have already seen it. Triangular, basically circular, triangular, it can be rectangle like, it can be boat shaped. So there are different shapes in the case of diatoms, right? In the case of diatoms. Now as I said, what are they? They are photosynthetic, right? What are they? They are photosynthetic. So if they are photosynthetic, definitely they have the chlorophyll. And you should know this thing. If you know it, very good. Otherwise just focus on this word, photosynthetic. So if it is photosynthetic, means it will have the pigments. Okay, so chlorophyll A, it is the universal pigment, right? Chlorophyll A, it is the universal pigment. So in all the organisms that are photosynthetic, right, that are photosynthetic, you will find chlorophyll A. What will you find there? You will find chlorophyll A for sure, because it is what? It is photosynthetic, right? It is photosynthetic. So chlorophyll A will always be there in the photosynthetic organism. So there you will have chlorophyll A, chlorophyll C, and xanthophylls are there. So bache, because of these xanthophylls, they have different colors. Okay, the color is because of pigment. One student was asking me na about the pigment, about the color. So I'm saying they will have different colors. Like if I'm saying that desmid is a golden algae, because of that appearance, and that appearance will be because of pigments. So in the case of diatoms, there will be chlorophyll A, there will be chlorophyll C, there will be xanthophylls. So which xanthophyle will be there? Fucoxanthin. Like in the case of brown algae, we have the fucoxanthin. In the case of brown algae, we have the fucoxanthin. So fucoxanthin is going to give the brownish kind of color. Okay, so different shapes are there. Diatoms can live in fresh water and sea water. Photosynthetic they are, so they will be having different pigments. Chlorophyll A, chlorophyll C, xanthophylls, right? So particularly fucoxanthin is very dominating there. That is why they have the different colors. Is that clear? Is that clear, bache? Is that clear? This point clear? Now the next point is the very important and the peculiar feature in the case of diatom is their structure, their cell wall. Right, their structure, their cell wall. Now let's talk about the cell wall of the diatoms. So when you talk about their cell wall, right, in the case of diatoms, you will see the structure in this form, right, in this form. But everything is written in NCERT. Everything is given in NCERT, and this is what you need to know for your NEET paper. But right now we have to understand the concept, so you better focus here, right? This point is very important from examination point of view. So the very important thing in the case of these diatom is what? Their cell wall. Now when you talk about their cell wall is having silica, right? Cell wall is having silica. This cell wall is not having cellulose. This cell wall is having what? Silica. Now when you look at it, okay, what's that? Okay, so when you look at it, just look at it. Don't you think that it is just like, you know, two covers? It is just like two covers. Like let's say we have a box here. Okay, we have a box here, right? So okay, 1 2 1 2. Can you see this? Can you see this? And here even you can see some demarcation. 1 2 2 parts, right? So now when you look at the structure of diatoms, their cell wall contains silica, and moreover when you look at the structure, so see the upper portion, the upper part of this, it is basically epitheca. What is it? It is basically epitheca. The lower one is hypotheca, right? It is hypotheca, and it looks like a soap box, isn't it? It looks like a soap box, isn't it? Isn't it? This structure, this arrangement, it looks like a soap box, right? So the upper one, epi means above always. What is the meaning of epi? Epi means above always. Hypo, this one is the lower one, right? So that is how the structure is, and here in between there will be a groove, right? What will be there? There will be a groove. So when it comes to the diatom, this point is very important. And what is it? It is their cell wall. Cell wall is having silica, and because of the presence of silica, it becomes indestructible, right? Indestructible means after the death, these diatoms, they are not going to, they will not get degraded, right? Like in a sea, basically, just imagine this is a sea, right? This sea is having so many diatoms are there. So after the death of the cell, right, obviously all the diatoms, because they are having silica, they will not get degraded. Because of the presence of silica, they will not get degraded, and because of that, what will happen? They will accumulate here at the bottom of the sea, right? It will accumulate here at the bottom of the sea. So see, first of all, just look at the structure, epitheca, hypotheca, there will be a groove in between, right? We are not concerned with the groove. We don't want to know about the groove. It is beyond our syllabus. Okay, so now we are saying that because it is silica is there, the cell, this structure will become indestructible, so they will get accumulated here, and because of that accumulation, what will happen, bache? Right here, after so many years, it is not a one-day process. After many years, you will get diatomaceous earth here. right, diatomaceous earth here. Right, it will be diatomaceous earth. You can also call it as diatomite. Right, and even you can use the word Kilsger. Right, it is also known as Kilsger. So basically in the exam they can ask you this question. Right, right, you can just note down, please note down. You can they can ask you this question in the paper, that who formed the diatomaceous earth? Obviously the diatoms. So it is also known as Kilsger, it is also known as diatomize, it is also known as diatomaceous earth. So first of all, please note down, this point is important. Okay, this point is important, please note down. Right, so Kilsger, diatomaceous earth, diatoms, so these are basically the accumulations. Right, these are basically the accumulations of the cell wall of diatoms. Okay, done, fine, right, sure, can we go ahead? Acha, Kilsger nahi, it's Kieselguhr, you're right. Done? Kieselguhr. Fine. Now, next point is, bache, when you talk about the diatoms, they do not possess flagella. Okay, they do not possess flagella. Only reproductive structures will have flagella, like gametes. Fine, only reproductive structures contain flagella. So when you know that when it is the reproductive structure, means the gametes here, right? So diatoms, they move by gliding movement. Basically they show gliding movement, right? Some mucilage will be secreted by these cells, and with the help of that it will move. Okay, with the help of that it will move. Okay, are you getting it? So they do not possess flagella. So basically immotile it is. Only reproductive structures like gametes, they will contain flagella, right? So it moves by a gliding movement. So there will be the secretion of some mucilage. With the help of that, these diatoms are going to move. Okay, with the help of that, these diatoms are going to move. So this is important. And now, bache, the next important point is, in the case of diatoms, they show, right, they used to show what? They show asexual reproduction mostly, right? And when it comes to the asexual reproduction, next point is also very important. Please listen to me very carefully. So when they show asexual reproduction, they go for binary fission, right? They used to do what? They go for binary fission. Now see, this is the structure of a diatom. Looks like a soap box. Okay? So when it is the binary fission, means it is going to divide into two. Bi means two, fission it is, right? Fission it is, there will be the breakdown. Okay, so here there comes a point when epitheca will remain epitheca, but moreover even hypotheca will become epitheca, right? Hypotheca will become epitheca, like this. So when it grows, when it divides with the help of binary fission, so eventually, bache, there is a decrease in the size of these diatoms, right? This is something important that you should understand. Fine, bache. So basically what is happening, these diatoms, when it comes to the asexual reproduction, they divide by a binary fission. So because of continuous binary fission, finally there will be a decrease in the size, right? There will be a decrease in the size, because there comes a point when hypotheca will become epitheca during that binary fission. So because of that, what is going to happen? The next generations, the daughter cells will be having very small size. So now how do they restore? How do they restore the size? Do you know about it? How do they restore the size? So they restore the size by forming oxospore. This is important. Restore size by the formation of oxospore. But this word is very important, right? They restore the size. How? With the help of oxospore formation. Basically they form the oxospore, means the cell structure here, the cytoplasm, here one protrusion will be there and that is going to form the oxospore. I will tell you how the oxospore will form, but right now just mention it that this word is related with the diatoms. From many years you are not going to get any question in the last 5 years. Right, in the last 5 years, I don't think that you will get a question from this particular word oxospore, but if you will check the paper of 2015-16, so in the AIMS paper, there is a one question on the basis of this oxospore. So I always used to tell it in my lecture, right? Right, so diatoms, they are going to restore their size with the help of the formation of oxospore, but how this oxospore will form? So this oxospore formation is related with the sexual reproduction. Okay, it is related with what? It is related with the sexual reproduction. Now you must be thinking that, ma'am, how? Right, so basically when unfavorable conditions are there, now they are decreasing their size. Obviously it is also a kind of unfavorable condition. So during unfavorable condition, right, during unfavorable conditions, these cells, they undergo the sexual reproduction. What will they do? Right, what will they do? Okay, so what will they do during unfavorable conditions? Right, bache, they can form the oxospore. So oxospore is nothing, it is the extrusion of protoplast. As I said, it is basically the extrusion of protoplast, and you can say that, yeah, basically the extrusion of protoplast or a kind of extraction of the frustule here. Right, expansion of the frustule here. Now you must be thinking that, ma'am, what is frustule? So I missed one word here. So when you talk about the diatoms, right, their cell wall, for their cell wall we use the word shell. Right, it looks like a soap box, and we also use the word shell or frustule. Right, we also use the word shell or frustule. Even it is important. I just missed it. Yes, even it is important. Right, cell wall or frustule. So when it is the oxospore formation, at that time the protoplast extrusion and the expansion of this frustule or shell will be there. That will form the oxospore. So basically here the gametes will form. The detailed procedure is not required. Gamete formation will be there. That gametes will fuse. You know that zygote will be there. That zygote is basically forming these. This zygote is nothing, this is basically that oxospore. Okay, the zygote is nothing, it is basically that oxospore. Is that clear? Is that clear? Yes, bache? Is that clear? So zygote is nothing, it is the oxospore. It is the oxospore, the rejuvenating spore by which the diatoms, they will increase their size. So I have added it here. So here you guys can see this shell or frustule. Right, here you people can see this shell or frustule. So for the diatoms, because the cell wall is containing silica, it becomes indestructible. These diatoms, after the death of the cell, right, the cell wall aggregate, the structures like that, they will aggregate at the base of the C. It will form diatomaceous earth, diatomite or kieselguhr. Right, bache? So here you guys can see mainly they divide by a binary fission. So now just look at the structure. So there comes a time now when hypotheca, when it is this hypotheca will also become the epitheca. So basically the smaller frustules will form with the time. So how are they going to restore their size? By forming oxospores. By forming oxospores. So basically oxospore formation is nothing, extrusion of protoplasm, expansion of frustule. Right, so gamete formation will also be there. This egg, this fertilization will occur, the oxospore will form. So this oxospore is nothing, it is the zygote basically, and then again it will divide. Fine, bache, again it will divide. So there are two ways. Clear? Clear, there are two ways. So small frustules can also form directly the oxospore or with the help of this. So here you guys can see this. Done, bache? Clear? Done? So just look at it. Where is the first step? Okay, so vegetative cell dividing mitotically, done. Mitosis is going on, done. Okay, so the size is decreasing, some environmental cues will also be there, right? Then what will happen? Where is third, first, second, okay, third. So see here the gamete formation is there, right? This is how in between these diatoms the gametes will form. One will form this sperm, another will form this egg, it will fuse. This is syngamy, the zygote is formed. This zygote is nothing, it is the oxospore. So this oxospore expansion will be there, new cell wall formation in the oxospore will be there, and then initial cell formation will be there, and this is how further it will grow. So any doubt here? Any doubt here? Right? Any doubt? So clear, the concept of oxospore is clear. I have seen many teachers, they don't explain oxospore because we don't get question on the basis of this oxospore. But actually, if you check the AIMS paper of 2015, if I am not wrong, you will find a question from it. Right? You are going to find a question from it. So that's why I am mentioning it here. Done, bache? So you can take a screenshot. Okay, you can take a screenshot. Okay? Fine. So now bache, the important points again. So obviously we are saying the diatomaceous earth will form, but what is the role of that diatomaceous earth? That earth is gritty. Like sometimes, you know, we do you know about that face scrub? Face scrub is having some, you know, some granules. It feels granulated, right? So this diatomaceous earth is also gritty. So it is used for polishing. And when you talk about the diatoms, they are the chief producers of the oceans. Okay, they are the chief producers of the oceans. They are photosynthetic. Obviously, photosynthetic, they are going to do what? They are going to fix that carbon dioxide. And moreover, bache, moreover, these diatoms, they are pollution indicator. Do you know it? They are pollution indicator. Pollution indicator. So, if there is the hyper growth of diatoms, if there is the hyper growth of diatoms, so it indicates pollution. Fine, it indicates pollution. Hyper growth of diatom indicates pollution, first point. And secondly, they are also known as the pearls of oceans. Why? Because of their silicified, right, cell wall, right? So chief producers of oceans, they are diatoms. Okay, diatoms. So see here, indestructible cell wall is there, right bache? So cell wall makes two thin overlapping shells which fit like a soap box on the outer surface. Diatomaceous earth will be formed. It is used in polishing, filtration of oils and syrup. So they are the chief producers in the oceans. That's all. This is all about the chrysophytes. And when it comes to the desmid, as I said, they are the golden algae. So in the case of desmids, you won't see, basically they will always grow in the fresh water. This is what we need to know. Okay, so obviously if it is not growing, it is indicating that water is polluted. Okay, water is polluted. Fine. So the next that we need to discuss is dinoflagellate. So kids, again revise it with me. Say CDESP, dinoflagellate, dinoflagellate. So the first one is chrysophytes, dinoflagellate, euglenoid, slime mold, protozoa. Chrysophyte, dinoflagellate, euglenoid, slime mold, protozoa. Clear? Chrysophyte, dinoflagellate, euglenoid, slime mold, protozoa. This is what you people need to remember. So when it comes to the dinoflagellates, so basically it is a biflagellated protista. It is having two flagella, right? Dinoflagellates, they are biflagellate, right? They are biflagellated protozoans. Fine bache? Fine bache? Okay. Dinoflagellates. Done? So it is also photosynthetic. Dinoflagellates, they are also photosynthetic protistan. It is biflagellated. It is having two flagella. We will discuss about it. And they are mostly marine. Few forms are in fresh water also. Bache, because of their color, mostly they are golden brown, right? They are golden brown algae. We also call it as golden brown photosynthetic algae. It is what? It is golden brown, right? It is golden brown. Okay, not simply golden, it is golden brown photosynthetic protista. So obviously if it is biflagellated, it is going to be motile without any doubt, right? So this is what we need to discuss, and I already told you that in the case of dinoflagellates, we will see zygotic meiosis, right? What will we see? We will see zygotic meiosis. So the main cell is haploid. Okay, so the main cell is haploid. So zygotic meiosis means haplontic life cycle will be there, right? Haplontic life cycle will be there. And bache, dinoflagellates, they are having different colors because of the presence of different pigments like chlorophyll A, C, xanthophylls will be there, no doubt, but they are having different type of colors, red, green, blue, like this. Here in this topic we will also talk about the red tides also, right? But these are few general points related to the dinoflagellates. Okay, related to what? Related to the dinoflagellates. Now bache, when we talk about the dinoflagellates, you already know the basic points. Now the points that can so the points from where the question can come, just note down. Okay, so in the case of dinoflagellates, they are also having the cell wall, right? They will also contain the cell wall. Cell wall will also be there, but here the cell wall is made up of cellulose. So cellulosic plates, stiff plates, cellulosic plates will be there, right? What will be there? Cellulosic plates will be there, right? So two plates. Again here also you can use the word epitheca and hypotheca, right? You guys can use the word epitheca and hypotheca here, but the important point is it is made up of cellulose. It is not siliceous. It is not made up of silica. Okay, it is not made up of silica. And bache, here in the case of dinoflagellates, there will be two flagella, but these two flagella, they will arise from a common point. Okay, they are going to arise from a common point. Let me show you this and this. Two flagella will be there, right? Two flagella that arise from a common point, right? That arise from a common point. One is longitudinal, one is transverse. Okay, transverse. So basically what is happening, bache? See, in the case of diatoms, this part we use the word we use the word sulcus for it, right? We use the word sulcus for it, and here this portion is considered as the groove. So when you talk about the dinoflagellates, so as I said, they are having two flagella. So these two flagella, they will arise from the common point, right? It is going to arise from a common point. So one will be there in this sulcus, another will be there in this groove, right? So these are two different flagella which are present in the case of dinoflagellate, and for this flagella, right, we also use the word heterocont. This is extra. You want to write it down, you can, otherwise you can drop it, right? One flagella will be like this only, another one can be tinsel, like it can have the hair, right? So this is about the flagella here in the case of dinoflagellates, right? Dinoflagellates. Clear, bache? Is that clear? Done? So two flagellas are there which are of different types, right? But they will arise from the same point. Done, bache? It will arise from the same point. Okay. Done? So heterocont means two different. One is smooth. I will write. So longitudinal sulcus, smooth it is, and this will be, obviously it is tinsel. It is having hair. It is having hair. Clear, bache? It is having what? It is having hair. Okay. So heterocont is the word. And yes, these are basically the girdles. Instead of groove, you can mention the word girdle. girdle or annulus or annuli. Done, bache. So now just look at it. The flagella is like this. The flagella is like this. The flagella is like this. Like this. One is transverse, another is longitudinal. It is hair-like, it is smooth. So now, because of this arrangement of flagella, right, they will roll and spin. If I talk about their movement, see, in the case of diatoms, I told you binary fission will be there. They do not have flagella. They are immotile, right? With the help of that mucilage, they will move. But here in the case of dinoflagellate, they are motile, right? They are motile, they are movable. In their case, movement is possible with the help of that flagella. So because of the arrangement of this flagella, so sometimes they will spin or roll. So rolling and spinning type of movement is there, right? In the case of dinoflagellates, rolling and spinning type of movement is there. What is there? Rolling and spinning type of movement. Rolling and spinning type of movement is there, and that is why they are also known as whirling whips, right? We also call them as whirling whips. Their movement is like that. What are they? They are whirling whips. Okay, what are they? They are whirling whips. Is that clear? Yes, bache, is that clear? Rolling and spinning type of movement. Okay. Fine. Sure? Sure? Priya, ma'am, cingulum. But cingulum is same like this annulus. Okay, whirling whips they are. Now in the case of dinoflagellates, there is one more point, and that is quite interesting. You know that their nucleus is very large. When you talk about their nucleus, now we use the word mesokaryon. Their nucleus is unusual. It is not like all other organisms. Nucleus, for nucleus we use the word mesokaryon. What is the word for the nucleus of these dinoflagellate? The word is mesokaryon. Means nucleus is large sized. It is quite prominent, right? Quite prominent. DNA is not covered with histone proteins. Okay, it is not covered with histone proteins. So mesokaryon it is, very large nucleus. Very large nucleus is there. Okay, very large nucleus is there. It is quite prominent, bache, right, bache. So DNA is without histone proteins. Okay, DNA is without histone protein, and in the case of dinoflagellate, there is no spindle formation at the time of division. Neither the centromere is there nor the spindle. Okay, there is no spindle formation. Okay. Okay. Done. And pigments are same. Photosynthetic, it is also photosynthetic. So photosynthetic it is, so definitely the pigments will be there. So there will be chlorophyll A, there will be chlorophyll C, there will be the xanthophyll, and yes, here the carotenoids are also there. Right, carotenoids are also there. Clear, bache? Clear, bache? So this is about the dinoflagellate. Now we will talk about the red tides. Okay, so here you can see, bache, all the important points are highlighted here. Two flagella, one is longitudinal, another is transverse, right? So they multiply rapidly, and toxins released by such large number can kill other marine animals such as fishes. So basically we have to consider few examples. One example of dinoflagellate is goniolix, right? What is the example? Goniolix. Write down, everyone. Right, one example is goniolix. But it is responsible for making red tides in seas. Right, it is responsible for making red tides in sea. Red tides means you can see that there is the hyper growth of this dinoflagellate. So if there is excess of growth of this dinoflagellate, so the sea will appear red colored, right, bache? The sea will appear red colored. So it is responsible for the formation of red tide. So it is a previous year question. It is something important, right? It is something important. Moreover, when you talk about the same goniolics, it can do one more thing. But it releases toxins. Okay, it releases toxins, and this toxin is very dangerous. This toxin is not going to destroy, it is not going to destroy the invertebrates present in the sea, but it will get accumulated in the shellfish. Okay, so now if human will eat that shellfish, because some human, you know that they eat the seafood, right? So if they will eat that seafood, so this toxin, it is not affecting that particular shellfish, it is not affecting that particular invertebrate, but it will get accumulated in their body. So whenever any human being will consume it, that human can get paralytic shell food poisoning, right? Paralytic shell food poisoning. Are you getting it? Paralytic shell food poisoning. So this toxin is basically, it is a neurotoxin. Imagine, it can cause the paralysis. Neurotoxin it is, right? It is basically, the particular name is sexitoxin. So I'm writing it here. It will cause no harm to invertebrate. You know na, invertebrates, vertebrates. So no harm to invertebrates, but will get accumulated in the body of shellfish, right? But will get accumulated in the body of shellfish. So humans will get what? Humans are going to get paralytic shellfish food poisoning, right? So in the case of humans, so if any human being is going to eat it, oh, I should write it properly, but I am not writing it properly, you please take a note of it. So in the case of humans, it can cause paralytic shellfish poisoning. Done? Sure? Done, bache? So goniolics, it is goniolics. Done? So it's a neurotoxin, bache. So paralytic shellfish poisoning will be caused. Okay. Done? Sure? Okay, fine. So here you guys can see this is the common point from where that flagella will grow. Okay, so heterocont is the word here. So one student was asking the cingulum. So cingulum is same like this annulus. See this. So epitheca and hypotheca, but this cell wall is made up of what? Cellulose. Okay, it is made up of cellulose. So mostly bache, it will reproduce by asexual reproduction in the same way, and even the sexual reproduction is also there. That will be, you know, isogamy and anisogamy. Isogamy means when gametes are same morphologically, right? Their size, shape, everything is same. Physiologically they can be same or different. And isogamy, when their size is not same, physiologically same or different, it can be, mostly they are different. Okay, mostly they are different. Okay, so this is about the dinoflagellate. So even their toxins can also kill the marine fishes, bache. Okay, so goniolics is the example, and along with that, you know, we have another example also, like one is noctiluca. So it shows bioluminescence. Do you know the meaning of bioluminescence? Bioluminescence, the living body, the living organism can emit light, right? The living organism can emit light. So that is the bioluminescence, the property of a living organism, that it can emit light. And why is it so? Because of the protein luciferin. Okay, because of the protein luciferin. So noctiluca, along with noctiluca, even goniolics, it also shows the property of bioluminescence. Okay, and it is not like that in dinoflagellates all will show. All the organisms are not going to show the zygotic meiosis. Some can even show the gametic meiosis also. It depends. Okay, so another example, like, and when it comes to the red tides, along with when it comes to this red tide along with goniolics, we have one more example of ceratium also. Fine, ceratium is also the example. It is also a dinoflagellate. Done, bache? It is also a dinoflagellate, ceratium. Done, bache? Okay, so this is about the dinoflagellates. So now we have to start what? We have to start the euglenoids, and trust me, it is very, very, very simple. Okay, euglenoids, they are very simple. So what do you know about the euglena? Yes, bache Priyanka, you are right, bache, very good. So what do you know about the euglena? Do you know anything about the euglena? Yes, anything related to euglena? Euglena or euglenoids, any euglenoids that you know? Yes, bache. Okay, so bache, when you talk about the euglena, so yes, mainly they are in the fresh water, but they found in the stagnant water, right? You will see them in the ponds, in the lakes, in the stagnant water you are going to find these euglenoids. So now when you talk about the euglenoids, we have one very common example, that is your euglena. Bache, euglena is basically a connecting link, right? It is what? It is a connecting link between plants and animals. Right, it is a connecting link in between plants and animals. Actually, what is the story behind? Some scientists, because of the, see, euglena can perform photosynthesis when solid food is not available, and when the sunlight is not available, they can even go for the solid food. So that is why we used to say in the case of euglena, we see the mixotropic nutrition, right? What type of nutrition is there in the case of euglena? The nutrition type is mixotropic. Right, mixotrophic, like it can be holophytic, plant-like, it can be holozoic, animal-like. It can be holophytic, it can be holozoic. Holophytic, plant-like, holozoic, animal-like. So some scientists, because of the presence of this photosynthesis, they kept it in plant kingdom. So earlier, euglena was there in the plant kingdom, but now in euglena, cell wall is also there. Right, now in euglena, cell wall is also there, right? Cell wall is also there. So basically, and even this euglena can eat the solid food. So scientists, they were confused where to keep it, in animal kingdom or in plant kingdom. So later on, it was kept in kingdom Protista, right? And it is considered as the connecting link in both. Clear, bache? Connecting link in both, right, bache? So when you talk about the cell wall, right, when you talk about the cell wall, so basically you will not say that there is a proper cell wall, right? You will not say that there is a proper cell wall. So because they were eating solid food, or proper cell wall was not there, so they kept it in animal kingdom. So I won't say that there is a proper cell wall, but they are having one covering that is known as pellicle. That is known as pellicle. It is important. It is important. In the case of euglena, 3 words are there that you need to know, that you should know. One is this pellicle, another is this metabole, and one is your pyrenoids. Yes, pyrenoids. These are the 3 words that you people should know. The very first is pellicle. So pellicle is what? It is the outer covering. But this outer covering is proteinaceous. What is it? It is proteinaceous. It is made up of proteins. So if it is made up of protein, it is flexible also, right? It is flexible also. So this point is important, that they do not have the cell wall, but they have a protein rich layer. Means outer covering is there. It is not just a plasma membrane, it is not just a cell membrane, but what do you have here? Pellicle, right? So it gives a flexibility. It is protein rich, it gives flexibility. Right, it gives flexibility. So right now just focus on these points. Mode of nutrition, I have already explained you. It is mixotropic, it is holophytic as well. Holophytic means plant-like, photosynthetic. Holozoic, animal-like, solid food will be ingested. Right, so that is why mixotropic. Now in the case of euglena, but they are also known as the spindle organism. Do you know that? Euglena is also known as the spindle organism. What is it? It is also known as spindle organism because of, obviously, because of its body type. Okay, and now this is a very famous question from this part. Euglena is having 2 flagella. It doesn't have 1 flagella, it is having 2 flagellas. Okay, How many flagella are there? In the case of euglena, how many flagella are there? 2 flagella. One is underdeveloped, not properly developed. It is like this, but another one will be the longer one. Okay, how many flagella will be there? There will be the 2 flagella. One is long, another one is short. One is long, another one is short. Done, bache? Done, bache? Okay. Okay, so now when it comes to the movement, I told you that you need to know the 3 words. One word is bache, metaboly. One word is metaboly. Do you know that what is the meaning of metaboly? Do you know that what is the meaning of metaboly? Anyone in the class? What is the meaning of metaboly here? Anyone? Give it a try. Just give it a try. G. What is the meaning of metaboly? Right? And focus here. The outer covering is pellicle, which is proteinaceous, which is flexible. So just relate it. What is the meaning of metaboly? Actually, bache, when it comes to the euglena, right, their body movement is also by contraction and expansion of their body. See, their outer covering is proteinaceous. It is quite flexible. Okay, Srinath, outer covering is proteinaceous, it is quite flexible. So how do they show the movement? They show the movement by creeping, right, by the contraction of the body, and for that movement we use the word metaboly, right? So they can undergo creeping through expansion and the contraction of body, right? They can undergo creeping movements are there. So creeping through contraction and expansion of body. So this is basically the metaboly. Clear, bache? This is basically the metaboly. So it is about the euglena having a mouth as well, having the pigments as well. For what? For the photosynthesis. Okay, for the photosynthesis. Clear, bache? Clear? So this is basically the mouth region, the gullet, the cytostome part, the reservoir part. Wait, I'll show you the diagrams also. So just have a look. Have a look. Okay, so see this. Just have a look here, bache. So this is the mouth region, right? Cytostome it is. Cytostome it is. And here, just near to this flagella, can you see this photoreceptor? Stigma here, red spot here basically. Right, photoreceptor which will receive the light, and here we will also have contractile vacuole. As I said, you will find it in stagnant water. You will basically find it in fresh water, right? So where will you find the euglena? You are going to find it in fresh water, mainly in the stagnant water, right? Still water, and it is basically the fresh water. So if it is in fresh water, it needs to balance its salt and water concentration as well. It needs to balance its salt and water concentration as well. So for the osmoregulation, right, this is the role of contractile vacuole. But individually they can even ask you the question. So contractile vacuole is playing role in osmoregulation, that is maintenance of salt and water, right? That is maintenance of salt and water. So rest all the organelles are there, chloroplast is there, stored carbohydrate is there, nucleus, nucleolus, flagellum is there, and this is the photoreceptor here, obviously, which will receive the light, and near to this is the stigma, which is also called as the eye spot. Okay, it is also known as the eye spot. Clear, bache? Clear, bache? Done? Done. Okay. Done. So stigma, eye spot, and here it is the photoreceptor. So both the things are, related somewhere with because stigma is also pigmented. So they are related with the photosensitivity. Okay. So it is associated there. That is all. This is what you need. And bache, this is something important you all can write down. We will even study about it in the chapter cell. So see, when I was showing you the structure of flagella, so at the base, right here, near to the base, what do we have? We have the basal body. Always remember it. See, it is a basal body. For basal body, we also use the word blepharoplast. Sometimes the students, they don't know the synonyms. right? They don't know the synonym. So please mark it. Like always the flagella arises from what? The basal body, right bache? See, this is the basal body, this is the flagella. Now it's a eukaryotic cell. I told you already, flagella will be having 9+2 arrangement. It is made up of microtubules, which are in return made up of tubulin protein, right? Which are in return made up of tubulin protein. And from where it arises, right? It always arises from the basal body having 9+0 arrangement, right? So basal body is also known as blepharoplast. Okay, it is known as blepharoplast. Clear? What is it? It is blepharoplast. Even in the chapter cell, we will read this word. Clear, bache? So next is the slime mold. So C, D, E is over. Next is S, right? Next is S. So what are we going to discuss? We will talk about the slime molds, just like fungi it is. This is what we were discussing now. That in the case of kingdom Protista, there is no boundary. It will show the organisms like animals, plants and fungi. So when you talk about the slime molds, you can also call them as Protestan fungi, right? We also call it as Protestan fungi, right? Slime mold it is. What is it, bache? Slime mold it is. Clear, bache? Slime mold it is. Done? Okay, any other doubt? Any other doubt? Yes? One more thing, bache. Okay, so it is the Protestan fungi, or obviously it is a fungi. Mainly they are associated with what? They are the decomposer. So consumer decomposer Protestan it is, right? Consumer decomposer Protestan it is. Okay, okay, slime molds. And yes, how can I forget? Bache, this is a question. This is a very important question. Now, do you know about the paramecium? Do you know about the paramecium? Paramecium? Yes? Anyone? Paramecium? You know that now, paramecium is also a unicellular eukaryotic organism belong to kingdom Protista. It's a ciliate, right? It's a ciliate. We will study it in protozoa. So now here, why am I mentioning this? Bache, because there is a PYQ. They can even ask you about this euglena and paramecium's division, right? See, in the case of this euglena, it will also divide by, right? Mainly in the case of euglenoids, you are not going to see the sexual reproduction, you will see asexual reproduction. Okay, here in the case of euglenoids, you are not going to see, right? You are not going to see the sexual reproduction. Majorly you will see asexual reproduction, and that is the fission. That is the binary fission. Now, it's a previous year question. It's a previous year question, right? So binary fission in the case of euglena, what is it? It is longitudinal, right? Now paramecium, that belongs to protozoa, right? There it will be transferred. So yes, you used to get such kind of questions in the paper. Okay, so asexual reproduction will only be there in the euglenoids. You are not going to find the sexual reproduction here, right? So majorly it will be fission. Different type of fission can occur. And yes, one more thing from the diagram. So here the word is not pyrenoids, they are the paramylum bodies. Paramylum bodies, or you can say that paramylon it is. Paramylon. Many times I make this mistake. I'm telling you honestly, pyrenoids and paramylum, I confuse these two words, right? Most of the time I confuse these two words, paramylum and pyrenoids, right? Just before your class, when I was just checking out the content, so at that time even I was like, this time I will not make the mistake, but again I did. So basically the word is paramylum body, it is not pyrenoids. Pyrenoids, you know pyrenoids also, but that we discuss in the case of algae, right? Here the word is, here you know na, pyrenoids, starch plus protein, that we discuss in the case of algae. Here it is paramylum bodies or paramylon. Actually it is the product of photosynthesis. After photosynthesis in the case of euglena, right, what will form? Paramylon, which will be stored in paramylon body. So it is the product of photosynthesis, right? This is your homework, you will check its composition. Fine, you are going to check its composition. Fine, fine, this is what you people need to check. Okay, pythenoid we discuss in the case of algae. Done. So slime mold, that is what we are going to discuss. Slime mold is very interesting, right? From this diagram itself, I will explain you many things. So consumer decomposer fungi it is, right? Protestant fungi it is, decomposer it is, slime mold, just like fungi. Now here, can you see these beautiful, beautiful structures here? Small globular kind of structures here. Can you see these structures here? Just have a look. Just have a look. In the case of slime mold, you people will talk about the spore formation. Here the asexual reproduction will be there. There is no binary fission here. Here we will talk about spore formation. And this structure is the sporangium. It's a very important character here. That's why I'm explaining it here only. So sporangium. Sporangium, structure in which spore will form. What is sporangium? It is a structure in which spore will form. Structure in which spores form, right? Sporangium. So here you will see sporangium. So sporangium, from this sporangium spores will release, each spore will again grow. So very interesting characters are there in the slime molds. In the slime molds you will see that the spore will be having, just a minute, it's right. So see, just listen to me very carefully. See, so this is the sporangium. What is it? It is the sporangium. Okay, sporangium. So here in the case of the sporangium, spores will form, that spores will germinate and individually, you know, slime mold will grow. Okay, all the slime molds will grow. Are you getting it? Are you getting it? Yes, bache. And I was saying something, yes. So when you talk about the slime molds, now you will see the vegetative structure is not having the spores, right? Sorry, it is not having the cell wall. So during asexual during unfavorable condition, these slime molds will also go for the sexual reproduction. So at the time of sexual reproduction, what is going to happen at the time of sexual reproduction? What will happen here in the case of sexual reproduction? Spore formation will also be there. Right, again the spore formation will be there. That spore will be having the cell wall. So you will see that normal vegetative structure is without cell wall. The spore is having the cell wall for the protection. So question can come from such points. Okay, question can come from such points, and this is what we need to focus here. In the case of what? In the case of slime molds. Fine, bache. In the case of what? In the case of slime molds. Right. So here, first of all, just have a look of few diagrams. Can you see the structure? Right, even if you will just look at these slime molds, they are not having they are not even having a proper structure, right? They are not even having a proper structure. It is looking like a mass. Okay, it is just like a mass. Okay, slime mold. So first of all, let's talk about their vegetation, bache. So let's talk about their habitat, bache. So decaying vegetation, common on lawns and moist fields. Right, if you will observe on the trees as well, you will see such kind of structure. Have you seen it ever? Have you seen any structure like this ever? Have you seen any structure like this ever? Yes, ever? Yes, everyone, just let me know in the chat section. Quick, 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 quick. Quick. Never. Just from now start observing it. Start observing such kind of things in your surrounding. You will find it, and obviously you will find it on the moist trees, on moist wood. Okay, you guys will observe it. So slime mold, decaying vegetation, common on lawns and moist fields. So one thing we can conclude that they are going to be sporophytic, isn't it? What is it? It will be sporophytic. What is it? It will be sporophytic. Right, right. It will be sporophytic. It is sporophytic. Okay, Okay, because it will feed on dead and decaying organic matter. They can be parasitic also. Even some dinoflagellates are also parasitic. Fine, bache. And now here in the case of slime molds, you will see characters like animals and fungi. I just told you now, animals and fungi. I will explain. Just wait. See, mode of nutrition is saprophytic, done. Plasmodium. See, here also we use the word plasmodium. Actually, what is happening, I'm repeating this point again because you guys can get question from this particular part. Somatic cells in the case of this slime mold, they are without a cell wall, right? Somatic cells in the case of this slime mold, they are without cell wall. They do not have any cell wall, right? They do not have any cell wall. No cell wall means animal-like character, isn't it? No cell wall means animal-like character, isn't it? Yes or no? Yes, bache, yes or no? Have you noticed it? So, somatic cell means without cell wall. Somatic cell is without cell wall. So, it is an animal-like character. But in the case of spore, like fungus, you will see the cell wall will be there, right? So, now I am saying that somatic cell is without cell wall. So, what is going to happen? These cells, right, although it is unicellular, they are going to form kind of aggregations, right? They are going to form the kind of aggregation. Aggregation means all together. So, under favorable conditions, see this word, just focus on this word, bache. Plasmodium, it's an aggregation of slime molds formed under favorable conditions. So, now, this is the most common mistake that many students used to do, right? It's the most common mistake. See, plasmodium. You know that we study this word in protozoans also, isn't it? In the case of protozoans also, we study this word. See, if it is plasmodium, right, like this plasmodium, so it is a malarial parasite, we know about it, right? It also belongs to kingdom Protista. It is a malarial parasite. It used to cause the malaria, plasmodium. We study it in group protozoans, right? In the case of protozoans, we study it, right, bache? But here, in the case of slime molds, when you talk about the word plasmodium, so plasmodium signifies aggregation, right? It signifies aggregation. Like in the case of slime molds, when conditions are in their favor, many cells all together, they will form some aggregations. Like normally, have you seen the slime? Many children used to play with it. Slime, you can form different structure, you can give it any shape, right? I have a very little cousin sister, she used to play with it. Slime, have you seen it? You can give it any shape, you can just stretch it, you can just form circles with it, whatever you want to do, you can do with it. You can play with it. Slime. So obviously such words are taken from the biology only, right? So slime mold it is. So just imagine some aggregated structure, some single cells all together, they are joining, right? Some single cells all together, they are joining and joining, right? They are joining and joining. Are you getting my point? Right? So together they will form some aggregated structure, and that aggregated structures will grow, and obviously then they will result in the formation of that sporangium, right? These aggregated structure can go for several fields, okay? They can even grow for several feet, right, bache? So what is going to happen under suitable conditions? Some aggregates will form that can grow and spread over several feet, and we will use the word plasmodium for it, okay? So plasmodium, right? Plasmodium it is. Done? And yes, can you tell me, wait. It will not form sporangium initially, but it can grow for several feet. So basically many cells all together they will form. Now bache, do you know one thing? Tell me, is this structure multicellular or unicellular? Tell me this thing. Is it multicellular or unicellular? Tell me quickly. Is it multicellular or unicellular? Quick. Quick. Is it multicellular or unicellular? Anyone? Multi it will be, because many cells all together they will form the aggregations, right? So basically we discussed two words there, pseudoplasmodium and simple plasmodium, right? Pseudoplasmodium, when that aggregated cells, their common walls when they don't get dissolved, right? See bache, slime mold is a very interesting organism. You will see many characters. I am telling you now, you will see plant-like characters, you will see animal-like characters, you will see fungus-like character. Very important fungus-like character is it can feed on dead and decaying organic matter. It is saprophytic. Moreover, it is forming sporangium. It is also a fungus-like character. But along with that, just have a look. See, I told you plasmodium will form the aggregation. It's true. So obviously that cells, they will form a kind of multinucleated condition, right? So this is the story of the favorable condition. So during favorable condition, it can grow for several feet like this, right? It can grow for several feet like this. Now let's say, so multinucleated condition means what will be the word? The word is syncytium here. The word is syncytium here. Okay, multinucleated condition means syncytium. So it can grow for several feet. So what is the word? The word is plasmodium. Sometimes also here also you also use the word pseudoplasmodium. Pseudoplasmodium. Pseudo means false. So at that time, right, at that time, what is happening? All these aggregated cells, their common walls will not get dissolved, right? Their common walls will not get dissolved. Okay, their common walls will not get dissolved. Are you getting my point? Right? Are you getting my point? So that is a pseudoplasmodium. Now, under unfavorable conditions, what will happen under unfavorable conditions? When conditions are not favorable, then only it will go for the sexual reproduction, right? It will only undergo the sexual reproduction when the conditions are what? Conditions are unfavorable, they are not suitable, right? The surrounding, the environmental conditions are not favorable. So what will happen? Then this plasmodium will form sporangium, the structure in which spore will form. Okay, the structure in which spore will form. So obviously these diploid cells, obviously here you know that there will be some cells, they will undergo the division, the haploid spores will form here. When the again when the conditions will become favorable, the spores will release and again they will germinate, right? That is the same story. That is the same story. Guys, stop sharing your contact number here. Stop sharing your personal number here. Please focus here, bache. Please focus here. Right? We are not, bache, free to take your classes on Sunday because we have committed that we will finish your syllabus. Obviously, if you want to study, then only we are going to teach. If you are not interested, then even we don't have time. We have other things to do as well. So either please be attentive or leave the class. You just have two things to do. Attend it, be attentive, or otherwise leave it. Okay. Done. So normally it will be a syncytium, plasmodium, right? Sometimes you know these aggregated structure, they in that aggregated structure the cells are going to have their cell walls, the cells are going to have their membrane, right? Then we use the word pseudoplasmodium as well. Okay. Okay, so see, under unfavorable conditions, sporangium, and this is how it grows. So if you talk about the characters, see, somatic cell is without cell wall. I told you, somatic cell is without cell wall. That is animal-like character. What is it? That is animal-like character. Next point is spore. Now these spores which are released, they have cell wall. Obviously they have cell wall, it is going to protect them under unfavorable condition. So spores have cell wall. It is a plant-like character, isn't it? It's a plant-like character. And these sporangium Formation of sporangium. It's a fungi-like character. Saprophytic nutrition, although it is also in the case of some animals also, but again, the sporangium mainly, it's fungi-like character. What is it? It's a fungi-like character. It's a fungus-like character. Okay. Okay. Done. Fine. So this is about the slime molds. So here you guys can see the structure, the life cycle basically, right bache? So just have a look. Step 1, large multinucleated cell which are diploid that glides along engulfing food particles. This is a feeding plasmodium. As I said, the word here will be syncytium, because all the aggregated cells, their cell wall is fused, now it has become multinucleate. So this is a mature plasmodium which is preparing itself to fruit, means sporangium formation, right? This net-like appearance. So have a look of this young sporangium. So sporangia where spores will form, right bache? So in step 4, 2n nucleus undergoes meiosis, right? Diploid nucleus is undergoing meiosis. Just a minute. Okay, here you people can see diploid. Oh, what is happening? Just a minute, kids. Okay. Fine, leave it. So diploid nucleus undergo meiosis. It is forming the haploid spore. Obviously, it is basically meiosis. It is gamete spore. Ultimately, we need to form, right? So haploid spores will form. They will germinate, right? These spores, after germination, they will form these amoeboid cell, right bache? They will become flagellated, then they will fuse, and that is how the zygote will form. So basically here it is zygotic meiosis. So in sporangium, diploid cell will undergo meiosis, haploid spores will be formed, they will release, but they are having the cell wall also. Bache, this is in general, right? In general, this is what you can see. Spores germinate. Whenever they will germinate, they will form some amoeba-like cells that will become flagellated, that will fuse, syngamy, zygote. This is the entire life cycle. Okay. Okay. Done. Okay. Is that clear? So amoeboid cells, they are also known as swarm cells because they will ultimately fuse and they are going to form the zygote. That's all. That's it. Fine. This is about the slime mold. This is about the slime mold. Clear, bache? Okay. So the next and the last from kingdom Protista is your protozoans. So what do you know about protozoans? That is very easy. What do you know about the protozoans? It's very, very, very easy, bache. What do you know about the protozoans? Yes, it's quite easy. And do you know the examples of the slime mold? If you know it, good. Otherwise, please, 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 please. Right? Otherwise, please take a note of it. Physarum, dictostelium. Dictostelium is your cellular. Physarum is your acellular. I have to check it. I have to cross verify it. But yes, you can note down. Okay. So next is the protozoan. But sometimes now we are not able to explain, right? We are not able to explain the difference in between Protista and protozoan, right? So Protista is a kingdom. Protista is what? Protista is a kingdom and protozoan is a group there. Okay. Okay. So in the protozoans, on the basis of locomotion, we divide it into different categories, right? We will talk about it. So see, they are unicellular. Obviously, they are the believed to be primitive relatives of animals because they are having more character like animals and they are heterotrophs. I already told you that these protozoans, they come under ingestive protozoans, right? Or ingestive Protista, right? Basically, they are having holozoic nutrition. So holozoic nutrition means animal-like character, right? Animal-like character. So in kingdom Protista, you have seen plant-like, animal-like, fungus-like character. That is why we said they do not have the boundaries. Okay, they do not have the boundaries. So this is what we need to discuss here. So when it comes to the protozoan, see, amoeboid protozoan, amoeba-like. Obviously, amoeba belongs to this protozoa, right? Amoeba is a protozoan. So many students, trust me, many students, they are not even able to explain it, that that amoeba is a unicellular eukaryote, right? Sometimes they answer that it is in kingdom Monera. It is not. It is amoeba. It is amoeba. It's a eukaryote, right? It's a eukaryote. So amoeboid protozoan. Fine, amoeboid protozoan, right? So here for locomotion, pseudopodia will be used, isn't it? Pseudopodia is present. Now flagellated protozoan, obviously here for locomotion, flagella is there. Ciliated will have cilia, and sporozoan, no locomotory structure. They do not have no true locomotory structure, right? So when it comes to the sporozoan, your plasmodium, which used to cause the malaria, right? Plasmodium, which used to cause the malaria, it is also the example of sporozoan, right? So it will live in the body of the host. Okay, so on the basis of what? On the basis of the locomotory organelles, locomotory structures, the protozoans are divided into amoeboid, flagellated, ciliated and sporozoan. So sporozoan is having no true locomotory structure. Is that clear? Sporozoan is having no true locomotory structure. Is that clear, bache? Yes? Done? So on the basis of it, it has been divided into these 4 groups. This is what you need to know. Ultimately, it's a eukaryote. So other characters will be same like eukaryotes. Okay, other characters are going to be same like eukaryotes. Fine. So, bache, pseudopodia. So obviously you will find these protozoans, amoeboid protozoans in fresh water also, right? In marine water also. So let me tell you the examples here, and you know about such examples. Okay, you are aware of them. One is amoeba. You know that. It's a fresh water protozoan. Amoeba, it's a fresh water protozoan, right? This amoeba is what? It is a fresh water protozoan. Okay, and one more example we have, that is entamoeba, right, bache? That is entamoeba, and you know that it is a disease causing protozoan, isn't it? It's a disease causing protozoan, entamoeba histolytica. So you should know about it, and this is again your homework. You have to write these points in the comment section after the class. Which protozoans are the disease causing protozoans? Okay, bache? Which protozoans are the disease causing protozoans? Okay, so amoeba, freshwater protozoan, entamoeba, disease causing protozoan. Entamoeba, disease causing protozoan. So this entamoeba, it used to live in the, obviously its host is what? It is monogenetic, single host is there, host is human, single host is there. So for single host we use the word monogenetic, right? That is only single host is there, right? And the only host is what? Human. So it lives in the large intestine of humans. It used to cause the diarrhea, right, bache? It used to form the cyst. So this entamoeba is having, you know, 2 forms, minuta and magna, right? The magna is basically the magna is the one which will cause the infection. So it will cause the the diarrhea, watery stools will be there. Okay, so it will be in the large intestine. So in detail we will talk about this in human health and disease, but it is your homework. Write down the name of the disease causing protozoan. Now when it comes to the flagella, there are so many examples. I'll tell you the disease causing flagellated protozoans. So one is trypanosoma. Right, that one is trypanosoma. So trypanosoma is the one which will cause the sleeping sickness, right? There are different species of trypanosoma. Trypanosoma gambiense, right? Trypanosoma brucei, Trypanosoma cruzi, which will cause Chagas disease. So trypanosoma is responsible for sleeping sickness. Again, bache, it is your homework. You have to write it down. Bache, not even in NEET, in other, you know, exams, you can say that some CUCET exams like that, right? In nursing exams, you guys can get question from this part. Okay, so you please note down, you have to write it in your notes. Leishmania, Kala Azar, right? Leishmania, Kala Azar, it's a protozoan which used to show the pleomorphism. It is having different shapes. Remember, I have already used this word in mycoplasma. Pleomorphism is shown by this Leishmania. It is having spindle shapes, rounded shape also. Okay. Okay, Giardia is also there. Ciliated one is your paramecium, and it's a very famous question. It divides by transverse binary fission. right? It divides by transverse binary fission. Okay, so these are important thing. And in the sporozoans, Plasmodium it is, malarial parasite it is. Okay, it is a malarial parasite. So this is about the protozoan, and here you guys can see the disease caused by protozoan. So African sleeping sickness, Trypanosoma bruzi gambiense, malaria is caused by Plasmodium, amoebic dysentery, and Amoeba histolytica. Amoeba histolytica. And please note down, one bacterial dysentery is also there. Bacteria also used to cause the dysentery. Bacterial dysentery is known as schizalosis. It is also known as schizalosis. It is caused by Shigella. The name of the bacteria is Shigella. Okay, so amoebic dysentery is caused by Entamoeba histolytica. Clear, bachche? It is caused by Entamoeba histolytica. So another leishmaniasis in the case of dogs, humans and rodents, right? Babesiosis, Babesia. So plant disease like phloem necrosis of coffee, Phytomonas. So these are different examples. Okay. Okay, so now let's revise it from the NCERT ones, and then we will be starting the, we are going to start the kingdom fungi. So see, after kingdom fungi, we will be talking about the viruses, viroids, prions, lichens, very simple, right? That is a very simple topic. Okay, so let's finish it. And bache, if you are thinking that teachers are going to finish one chapter in 3 hours, it is not possible. It is literally not possible. Right, we really don't want to finish your syllabus. We want you guys to understand your syllabus first, right? Obviously, see, we are just taking 2 lectures for one chapter. Some chapters are too lengthy. Okay, so we have increased the duration. Mark my words. If you are willing to watch the one shots now, where the chapter is finished in 2 hours, 3 hours, so you can only understand from that chapters if you know something about that chapter. Okay. If you know something about this chapter, then only you will be able to understand it. Otherwise, it is of no use to watch that one shot and then go for the NEET examination. Okay, so you should know about that chapter. See, whenever we will be in the month of March or April, at that time we are also going to do the same things, the quick revision sessions. But before that, you need to understand the syllabus, and we have sufficient time. And let me tell you one good news as well, that for class 12 students also, because we are getting these messages also. So from class 12 students, I am going to, for biology, I will start the separate lectures as well. Okay, I am going to start the separate lectures for biology as well. For class 12th biology as well. As of now, we are focusing on 11th syllabus now. So parallelly, we will take it 11th and 12th. We'll let you know once I'll start. Fine. But show some sincerity. Be serious about this. Right, even if you are not interested in watching the live lectures, after the class you guys can increase the speed and you can watch it, and you can just note down the important things. It is important. Finish your syllabus properly, right? And this batch is for those who cannot afford any batch, or they want the quality education. They are not finding the teachers that can solve their problems. Yeah, they are not resonating with them. So please start preparing. Okay, so now let's revise it from NCERT once, and then we will have a break. Okay, we will have a break for 10 to 15 minutes. Then we will go for fungi, viruses and all. Even you take your dinner, I'll take mine. Done? No problem at all. Now please focus here. So kingdom Protista already covered. All single-celled eukaryotes, they are placed here. So boundaries of this kingdom are not well defined. Okay, so this can be asked as a question in the paper. They can be photosynthetic to one biologist. So they are putting it in plant. To another, it can be an animal. So in this book, it includes chrysophyte, dinoflagellate, euglenoid, slime mold and protozoa. That is why the trick here is CDESP. Okay, that is why the trick here is CDESP. Isn't it? Isn't it? So members of Protista are primarily aquatic, right? So they are going to form link with the others dealing with plant, animal and fungi. So I have given you many examples here. Okay, so being eukaryotes, they will having the cell body contains a well defined nucleus, proper membrane bound organelles will be there, flagella and cilia will be there, and flagella cilia will be having 9+2 arrangement, right bache? Asexual and sexual, both the reproductions are there, right? Right, so chrysophyte, which includes diatom and golden algae, so you know that they are in fresh water as well as in marine, microscopic, and they float passively in water current. So if something is flowing passively in water current, we use the word plankton for it, right? We use the word plankton for it. So mostly they are photosynthetic. That is why for diatoms we even use this chief producers of the oceans, and they are the pearls of the oceans, right? They are the pearls of the oceans. Clear bachche? So thin overlapping shells are there. So for that cell wall we use the word shell or frustule, right? Shell or frustule, isn't it? Isn't it? Right? So it is responsible for making the diatomaceous on earth. Again a previous year question. Fine, it is again a previous year question. Clear bachche? Clear? So dinoflagellates, mostly marine and again photosynthetic they are. They can have different colors because of the different pigments. So cell wall is made up of cellulose here. So cell wall is having stiff cellulosic plates. This is what you need to remember. Okay. So two flagella are there, but both the flagellas are different. That is why we use the word heterocont for it. Right? Heterocont for it. Is that clear, bache? So goniolics example is given here for the red tides also. Even goniolics used to produce the toxins. Next is your euglenoids. Majority of them are in fresh water and they are in stagnant water basically. Right? Two flagella will be there. One short, one long. Right? So they are photosynthetic in the presence of sunlight. They will go for the photosynthesis in the absence of it. Right? Holozoic nutrition will be there. That is why they are mixotropic. Clear bache? So they behave like heterotrophs by predating on another smaller organisms. Clear bache? Next is slime mold, saprophytic protista, right? Grows on dead and decaying leaves and twigs. Fine bache? Aggregation is known as plasmodium. Under unfavorable condition it will form sporozone, and this is what we have already explained. So now there will be a break. Okay, so your homework is you have to write a short note on all the protozoans which used to cause the disease. Okay, so there will be a break till 8:30. Yes. Right? And after that break, fungi, viruses. This is what we are going to discuss. Okay, fine. So see you after the break. Till then, please have your dinner. Hi kids, hello, hello, hello. Hi, hi, hi. How are you? Hi, hello, hello, hello. How are you? So done with the dinner? Yes. Are you all done with the dinner? Okay. So can we start the next kingdom now? That is kingdom fungi, and then viruses, viroids, prion. Very easy it is. It will hardly take 15 minutes to discuss that, and then the chapter will be over. And because tomorrow Shreyas sir is taking a marathon, so, and even I have a special class. Let me tell you once again kids, so tomorrow at 4 p.m. I will be taking one special class on Unacademy platform, right? And it is a free special class for all of you. Okay, it is a free special class for all of you. So what you people need to do, you just need to, right, you can just download the Unacademy app, and there you guys can check my special class, right? And here in this special class we will be discussing the questions of cell cycle and cell division, right? This is what we are going to discuss. So what will we do? The questions of cell cycle and cell division will be discussed here in this particular class. Okay, so you all can be the part of it. Obviously you are going to learn something. Done bache? Done. So tomorrow, maybe because Shreyas sir's marathon is going to take some time, so I will not take the question practice session tomorrow. So let's keep it on Tuesday at 4 p.m. or 3 p.m. We'll let you know about the timings. Okay. Okay. So this is what we will do. So on Tuesday, the question practice session of living word and the biological classification will be there, and then on Wednesday there will be the marathon of the plant kingdom. Fine bache, there will be the marathon of the plant kingdom. Clear? This is what we are going to do. So now let's talk about the kingdom fungi, and tell me, what do you know about the kingdom fungi? What do you know about the kingdom fungi? Yes, bache, what do you know about the kingdom fungi? So very first thing that you all should know, that you know that R.H. Whitaker, right? R.H. Whitaker introduced this kingdom fungi. He kept such organism in a, completely different kingdom fungi. This is what we have discussed. And now when you talk about the kingdom fungi, first of all, please note down these words. So study of fungi is known as, right, study of fungi is known as mycology. What is it, bache? Mycology. When you talk about the fungi, this organism is very interesting, right? Because see, first of all, they are eukaryotic. Obviously, they are eukaryotic. They are achlorophyllous. What is it? It is achlorophyllous. What is the meaning of being achlorophyllous? That is, it is a without chlorophyll. So if there is no chlorophyll, there is no chance of photosynthesis, isn't it? There is no chance of photosynthesis at all. So this is what you need to note down, that it is achlorophyllous means it is without chlorophyll. So 110% there is no chance of photosynthesis. So obviously it will be non-photosynthetic, isn't it? Non-photosynthetic. Done, bache? So you know that mostly the fungi is what? It is multicellular, isn't it? It is multicellular, but we have one exception. The exception is yeast. Remember this? Yeast, right? It is the unicellular fungi, and it is not just the yeast that is the unicellular fungi. We have another example also. Okay, and that is your homework. You have to tell me it in next class. Okay, it is your homework, right? So no doubt fungus is multicellular, but we have an exception of yeast, right, which is used for the baking, which is used in baking industry, in the brewing industry, and yeast is unicellular. It belongs to the class Ascomycetes, but there are other examples also where you will see the fungus is what? It is unicellular, right, bache? So eukaryotic, achlorophyllous, non-photosynthetic, multicellular it is, and bache, it can grow anywhere. So if it can grow anywhere, so we use the word cosmopolitan in distribution, right? Cosmopolitan in distribution, which can grow anywhere. So usually it prefers the moist and the humid conditions, but on the trees, right, on dung, on wood, right, you can find the fungus. And yes, sometimes on the body of a living organism, right? And even within a living organism, even within a refrigerator also the fungus can grow. And you know what, fungus can even grow on nails. Literally, it can grow anywhere, on leather, on nails. Are you getting it? Are you getting it? So what are we discussing? We are talking about the kingdom fungi, right? What are we discussing? We are talking about the kingdom fungi. So the study of fungi is what? It is mycology, right bache? Heterotrophic organisms are there. Heterotrophic organisms are there, right bache? So in their case, what are we going to get? What are we going to get? There will be no chlorophyll at all. There will be no chlorophyll at all, right bache? So if it comes to the nutrition, obviously heterotrophic will be the word if they are non-photosynthetic. And here also mostly you will see saprophytic, right? Mostly you will see what? Saprophytic. Now I don't think I need to explain these words. You know the meaning of saprophytic, which grows on dead and decaying organic matter. Or they can be parasitic, right? They need the host body, they will take their nutrition from them. And they can be symbiotic, they can live in the symbiotic association as well. So can you tell me the example of the fungi? Any example where fungi is living in the symbiotic condition? Do you know any example where fungi is living in the symbiotic condition? But we will talk about the structure, but as of now just answer this question to me. Any? Yes. Any example where you all can see that yes, the fungus is in symbiotic association. We have very common examples. Lichens, mycorrhiza. Lichen is there, mycorrhiza is there. These are the two common examples where you will see that fungus is in symbiotic relationship. And what is the meaning of a symbiotic relationship? Where both the organisms are benefiting each other. Right, where both the organisms are benefiting each other. So one is the lichen and another is what? It is mycorrhiza. We will talk about it, right? So these are the characters. And moreover, if I talk about the fungus is thalloid. Do you know that? It is thalloid. So thalloid means thallus-like, means body is not differentiated into proper root, stem and leaf. Okay, body is not differentiated into proper root, stems, proper root, stem and leaves. Just a minute. And leaves. Are you getting it? That is what fungus is. It is thalloid. What is it? It is thalloid, right? And moreover, bache, if you talk about this fungus, like if you talk about its structure, right? Can you tell me anything about the structure of the fungus? Yes, anything about the structure of the fungus? Anything that you know about the structure of the fungus? Yes, bache. They show the variety, they are cosmopolitan, I told you already, but what else about their structure? Anything? Yes, bachcho, please show some energy in the chat section. Let's interact, bachche. We can finish the class till 10 p.m. But you have to support me for that. Hyphae, very good, bachche. Hyphae, exactly. So let's talk about the features of the fungus. So first of all, see, mostly they are terrestrial. Mostly what are they? They are terrestrial. Okay, they are terrestrial. But fungus need warm and humid conditions. What type of conditions are required? Warm and humid, right? What type of conditions are required, bache? Warm and humid conditions are required. And as I said, it can grow anywhere, right? It can grow anywhere, like on dung, on wood, right? On dung, on wood, on animal body, within animal body, even on nails and on hair, right? So they can grow anywhere, dung, wood, animal body, nails, hair. Obviously plant body means wood, hai na bache? It is the part of that plant body, so it can grow anywhere. Okay, it can grow anywhere. So I'll quote this example here because it is given in NCERT. So the fungus which can grow on dung is known as coprophilous. What will be the word for it? That fungus will be the coprophilous fungi, right? That fungus will be the coprophilous fungi. We will discuss the examples. Like we used to say na, rhizopus is a bread mold. Same way, mucor is the dung mold, right? Mucor is the dung mold. So this is what you need to mark. So mostly it is terrestrial fungus, right? Worm it is, mostly it is terrestrial fungus. Worm it is, humid it is, it can grow anywhere, like on dung, on wood, on animal body, on nails, and on here. So basically what is it? It is the coprophilous fungi, right? What is it? It is the coprophilous fungi. Is that clear, bachcho? Is that clear? Yes? Is that clear? Okay, so this is what you people need to remember. Now when you talk about its structure, see, its structure is, when we talk about the structure, first of all you need to know that fungus is haploid. Do you know about it? That fungus is what? Fungus is haploid. What is it? It is haploid. Right? It is haploid. Right, bache? Haploid. Main plant body is going to be what? It is going to be haploid. Now in the case of fungus, what is the story? We will see some thread-like structures. Right? We are going to see some thread-like structures. Right? Some thread-like structures. So these thread-like structures are known as hyphae. What is it, bache? Hyphae. Now obviously fungus body is not made up of one hypha, second, third, fourth, fifth, like this. No, of course not. So it is made up of network of hyphae. There will be so many hyphae, which will make it, right? And for that so many hyphae, we will use the word mycelium, right? Which word are we going to use for it? We will use the word mycelium. Okay, which word are we going to use for it, bache? We are going to use the word mycelium here. Is that clear? Yes, bacho, is that clear? Yes or no? Tell me, yes or no? Huh? Is that clear? If there is any doubt, just let me know, bacho. Just let me know if still there is any doubt. Okay, so main body is haploid, not differentiated into root, stem and leaves, right? They are mostly multicellular, and the network of hyphae is going to form the fungus body, which is mycelium. Now when you talk about the hyphae, there are two words that we need to understand, right? That there are two words that we need to understand. One is aseptate and cenocytic. One is aseptate and cenocytic. And the second word that we have is septate, right? One is aseptate and cenocytic, and another word that we have is septate, right? What is it? It is septate. Now see, just look at it. Now when you talk about these hyphae, see, maybe there is no, like let's say you know that it is a multicellular structure. We have the example of the yeast. Right? We have the example of the yeast, which is an exception, which is unicellular. Right? Now let's say there is one cell, there is another cell, there is another cell, another cell. Now if there is a cell wall, or you can say that if there is a cell membrane in between, then obviously their nucleus will remain separated. Right? So you can say that here the things are, it is a multicellular structure, and each, right, it is made up of 4 cells. Let's say it is a multicellular structure made up of 4 cells, like this. This is what you are going to say. Now when you talk about the word septa, septa means this division, septa means this boundary, this wall, right? Whenever you use the word septa, so septa means this boundary, this wall, right? This wall which is dividing it into 2 parts, into 2 compartments, like this. So now septa, right? You got it, bache? What is the meaning of septa? Right? So now it is aseptate, means it is without septa. It is without septa. So here in the case of hyphae, the structure is without septa, there is no boundary, there is no middle wall. So obviously it is going to be multinucleated, right? It is going to be what? It is going to be multinucleated. Right, bache? Right? Without septum it is multinucleated, and you know that if it is multinucleated, we will use the word cenocytic, isn't it? We will use the word cenocytic, isn't it? It is multinucleated, it is cenocytic. Now the another thing is septate, right? Another word is septate. So septate means there will be these boundaries, these walls. So if such boundaries are there, so each structure will look like this. It will be uninucleated. So if it is septate, it means it will be the cells here will be uninucleated, isn't it? It is having so many nuclei, but individually they are present, isn't it? So the structure will be like this. So aseptate and cenocytic, it will be septate. It will be septate. Now one more thing is there. Now bache, in this septa, okay, here in this septa, in between there is a pore. Okay, here in this septa there is a pore in between. This pore can be a simple pore. I'll quote the examples as well. This pore can be a simple pore, or there is another possibility that this pore can be like this, like it can have swollen ends, right? It can have swollen ends, right? Am I repeating this again? It is a hyphae. It is the, see, hyphae is going to make, the hyphae is thin filament like structure which is going to make the structure of the fungi. So many hyphae together, they are forming a network, and for that network we are using the word mycelium. This is what I just said. Now here if it is aseptate, so it will be multinucleated like this. If it is septate, so the condition is going to be like this. Okay, now my point is, in between these septa, in between these boundaries, there are pores present. This pore can be a simple pore like this, simple opening it is. This pore can have the swollen ends like this, right? Just have a look, bache. This pore can have the swollen ends like this, and when it is having these swollen ends, right, some barrel kind of shape, right? So then you will use the word dolipore septum, right? Dolipore septum for it. Which septum it is? It is the dolipore septum then. Dolipore septum then. Now, in the case of fungus, right, there are different classes. In the case of fungus, you need to talk about the different classes. So there is a trick for it. Okay, there is what? There is a trick for it. Like Phycomycetes, Ascomycetes, Basidiomycetes, Deuteriomycetes. Okay, Deuteriomycetes. Are you getting it? Are you getting it? So there is a trick for these classes, and you have to remember it in same sequence. It is pineapple with dates. What is it? It is pineapple with dates. So P stands for Phycomycetes, A stands for Ascomycetes, B stands for Basidiomycetes, D stands for Deuteriomycetes. This is the sequence, and in this sequence you have to remember it. All of you, are you getting it? This is the sequence, and in this sequence you have to remember it. So what are we saying? Phycomycetes, Asco, Basidio, Deuterio. Phycomycetes, Ascomycetes, Basidio and Deuterio. Now bache, this Phycomycetes is also known as algal fungi. It is also known as algal fungi. It's quite primitive. So that is why we used to call it as algal fungi. What is the name here? The name here is Phycomycetes. What is the name here? The name here is Phycomycetes. Now in this Phycomycetes we have two things. One is oomycetes, another is zygomycetes. Firstly it will be zygo and then it is oo, right? So here you have two things. One is zygomycetes and another is oomycetes. So in NCERT it is not given in this way, right? Right, it is not given in this way that Phycomycetes is further having zygo and oo. So overall we talk about the Phycomycetes only. Okay, but this is how it is placed. Now when it is the Ascomycetes, we even call it as sac fungi, because here we will talk about a sac-like sporangium, which is ascus. So it is Ascomycetes, or yeast belongs to this. Now when it is Basidiomycetes, it is considered as these members of Basidiomycetes, they are considered as, you know, some advanced, they are quite advanced, they are very good decomposers of wood. So we used to call it as club fungi. Okay, club fungi. And when it comes to fungi, Deuteriomycetes, it is imperfect fungi. It is also known as fungi imperfecti. Okay, it is imperfect. It is imperfect fungi, fungi imperfecti. Again, I'm repeating. Kingdom fungi having different classes: Phycomycetes, Ascomycetes, Basidiomycetes, Deutero. Phyco, Asco, Basidio, Deutero. Phyco is quite primitive. We also call it as algal fungi. It is having Zygomycetes and Oomycetes. Because in NCERT it is not given in detail. So overall we are going to talk about Phycomycetes. Ascomycetes, some ascus, some sac-like structures are there. So we are calling it as sac fungi. Basidiomycetes, the members of this particular class, they are very good wood decomposers. We also use the word club fungi for it. Mushrooms basically belong to this particular class. Deuteromycetes, imperfect fungi. What is the meaning of imperfect fungi? That this fungus is not having any sexual stage. We will only see the asexual stages here. We will only see the asexual stages here. Now you must be thinking that ma'am, why are you talking about it? You were talking about the hyphae, right bache? So in the case of hyphae, because I need to give you the examples, so that is why I am starting it from this particular portion. Now you know the sequence. Pineapple with the dates, pineapple with the dates, Phyco, Asco, Basidio, Deutero. So now related here with this part. Now when you are talking about the aseptate and cenocetic, you will discuss the members of Phycomycetes will have the such type of hyphae, right? The members of Phycomycetes will have such type of hyphae, and when it is septate, so Asco and Basidio will have such type of hyphae. Is that clear, bache? Asco and Basidio will have such type of hyphae. Is that clear? Is that clear? Yes, bache? Is that clear? Sure? Sure, sure, sure. Done? Okay. So in the fungi, you know, there are different members here in the fungi. You will see different members here in the fungi. Like we'll talk about the organisms which are playing role in, obviously yeast, which will play role in baking and brewing industry because of fermentation. You will even talk about edible fungi, right? You will even talk about non-edible fungi like toad stools and all, right? We will discuss it one by one. Mushrooms are edible fungi. We are going to talk about it. So fungi is very diverse, right? In class 12th, when we discuss the chapter biodiversity and conservation, right? So we see that in the case of fungi, the species richness is there, variety of species are there, right? So many species are there. Are you getting my point? So many species are there when you talk about the fungus. So these are some basic structures. So I hope now it is clear. So when it is simple pore, yes, you can even take this example. Simple pore is present in the case of Ascomycetes, and dolipore septum is in the case of Basidiomycetes. Of course, there are some exceptions also like rust and smut, but again, in general, Basidiomycetes will have dolipore septum. So, bache, up to this part, all clear? Do you have any doubt? Guys, if there is any doubt, please let me know. If there is any doubt, please let me know. Are you sure? Are you sure? Yes, kids, are you sure there is no doubt? Okay, so this is about the structure. Now, when you talk about the reproduction, there are different ways. Again there are different ways, and it is important. So, first of all is vegetative reproduction, right? Very first thing is vegetative reproduction. Second is asexual reproduction. Right, the first is vegetative reproduction, second is asexual reproduction, third is sexual reproduction. So question can come from this part, so kindly listen to me carefully. So when it is vegetative reproduction, so obviously here you will include fragmentation, right? You will include fission here, you will talk about the budding here, right? You are going to talk about the fragmentation, you are going to talk about the fission, you are going to talk about the budding there. So now when the point is fragmentation, fragments, right, fragmentation. So basically what is happening here, bache? Here in fragmentation, what is happening? See, you know that the thallus will be there, made up of mycelium, right? The thalloid it is made up of mycelium. Usually na fungus is microscopic. When see, two phases are there. One is vegetative, another is reproductive. Reproductive, when there will be the reproductive spore formation like this, you know it, right? So I'm saying that, so basically when it is in vegetative phase, it is microscopic. You will not be able to see it with the naked eyes, but slowly when it will start growing, when it will start attaining its maturity phase, then obviously the kind of visibility will come, right? I'm not saying that all the fungi are microscopic. You know that we can see it at least when it grows, right, when it is in the reproductive phase. So now you know that when you talk about the thallus, it is thalloid, not structure is not properly differentiated. You cannot say that the root, stem and leaves there, but yes, right, some thallus, some undifferentiated structure is there. So now this undifferentiated structure is made up of hyphae, it is made up of mycelium. So now let's say some of it is getting separated, right? So obviously if it will fall down, it will grow, again it will grow into a new fungus. So this is the fragmentation, right? Some fragments of that mycelium will get separated and they will grow into a new fungi. Very simple it is. Then comes the fission. One cell will break into two. This is ultimately the fission. One cell will break into two. This is ultimately the fission. So in the case of fungi, this is also a type of vegetative reproduction, the propagation, where one cell will divide in this way. Then comes the budding. So you will see it in the case of yeast. Yeast is like this. So in the case of yeast, you will see some, you know, irregular buds. Some buds will appear. These buds will get separated and then they will grow into a new organism. Right? So this is what? This is the vegetative reproduction here. Right, bache? This is the vegetative reproduction here. Right? So buds are nothing, they are just the outgrowths. Okay, they are just the outgrowths. Now bache, when it is the asexual reproduction, see, for the sexual reproduction and for the asexual reproduction, ultimately there will be the spore formation. What is going to happen here, bache? Ultimately there will be the spore formation. What will be there? There will be the spore formation. So we will talk about it, but before that you should know that which spores will form here. Okay, so they can ask you, right, they can give you in the question, in the final NEET examination, you guys can get a question like this. They can ask you that out of the following, which spore is the result of, right, which spore is the result of sexual reproduction, which spore is the result of asexual reproduction. So yes, question like this can come in your paper. So when it is the asexual reproduction, there are some spores. Zoospore, you can take a note. Sporangiospore, they all are the result of asexual reproduction. Conidia basically. Chlamydiospore basically. Oidea, right, they are the result of asexual reproduction, right? Zoospore, sporangiospore, conidia, chlamydospore, right, bache, oidea, these are the result of what? These are the result of what? These are the result of asexual reproduction. So now when it is the sexual reproduction, you have ascospore. We will talk about the fruiting bodies here. You have oospore, right? So oospore, especially in your Phycomycetes, ascospores, and then comes the basidiospore, right? Then comes the basidiospore. So oospore in Phycomycetes, ascospore in Ascomycetes, basidiospore in Basidiomycetes, and Deuteriomycetes, because it is without any sexual reproduction stage, so obviously it will not have these spores, isn't it? It will not have these spores. So guys, please take a screenshot of it for the quick revision. Quick, just take a screenshot of it for the quick revision. Quick. Quick bache, quick. Done? Done, bache? Yes? Sure? Okay. So now let's talk about the asexual reproduction. right? Let's talk about the asexual reproduction. So बच्चे, this is important for you. You should know that which spore is the result of asexual reproduction, which spore is the result of sexual reproduction. That is important to know. Okay, that is very important. So now when it is asexual reproduction, I told you about the zoospores, isn't it? So बच्चे, zoospores, the word is zoo. So the word zoo is associated with the animal, isn't it? The word zoo is associated with the animal. So ultimately these are the spores which are having flagella, right? They are the spores having flagella. So if flagella is there, so it simply means it is motile. Motile means movable, means animal-like character, right? It is having animal-like character. So there will be some structures, right, in which these zoospores will form. Are you getting it? There will be some structures in which these zoospores will form, and that structures are known as zoosporangium, right? They are known as zoosporangium. Are you getting it? So these are the zoospores. Even in the case of algae, we are going to talk about these zoospores, right? Even in the case of algae, we discuss these zoospores. So this is about the zoosporangium, zoospore. So bache, they are having flagella. It can be 1, it can be 2, it varies, right? It can be 1, it can be 2. The number of flagella is not fixed. Fine bache, the number of flagella is not fixed. Okay, so it can be uniflagellated, it can be biflagellated. Simple. Done bache, simple it is. Fine, fine, fine. So obviously they will grow, they will form the new mycelium. Okay, they will grow, they will form the new mycelium. So this is the one example, that is the zoospore. So second is the sporangiospore, right? Second is what? Second is basically the sporangiospore. So now in the case of sporangiospore, it is something like this, right? The structure is something like this. So see, it is sporangiospore. So obviously it will form within a structure, that is sporangium. So it is a kind of non-motile. The spores will be non-motile. They will be thin-walled, right? It is non-motile. It is thin-walled spore which formed endogenously. Endogenously means inside that fungus. Endogenously it will form, right? So it is non-motile, thin-walled, right? And it will form endogenously. Are you getting it? It will form endogenously. Third category is bache conidiospore, conidia, right? So actually what is happening, see, these sporangium is the structure in which spore will form. So obviously in the hyphae, in that mycelium, there will be some branches over which these spore forming structures will form. Over which this spore forming structures will form, and within that spore forming structure, these spores will form. Okay, these spores will form. Okay, so here same is the case here in conidia. Now in conidia, in the case of conidia is also the result of asexual reproduction, and bache, this is important. Okay, this conidia part is important. So see, like this. I think you all have seen a structure like this, isn't it? Isn't it? You all have seen a structure like this. So C for conidia, C for chains, and it is forming exogenously, right? It is forming exogenously, not inside a structure. But look at this, this branch is conidiophore. Okay, this branch is conidiophore, the branch over which these conidias, they will grow. So see, they will form in a chain like fashion, right? Externally they will form in a chain like fashion. This is conidia, and this branch is conidiophore. So this is a very common example that you will see in penicillium, right? You will see this such kind of, you know, conidia in the case of aspergillus. These are very important fungi. These are very important fungi, right? They are basically having economic importance. Okay, so this is conidia. Okay, bache, this is conidia. They are having different, right? Different shapes are there. You might have seen that. This is the one way. You know, some another ways are also there in which these conidia will form. You know, that branch can be like this, and then, okay, then, are you getting it? So these are different ways. Clear, bache? Clear? Okay. Okay, so this is what? This is conidia, right? It is conidia. Is that clear? Yes, everyone tell me, is that clear? Okay? Sure? Fine. So fourth one is your chlamydospore, right? The fourth one is your chlamydospore. So it will form under unfavorable condition, right? It will not form in favorable condition. So under unfavorable condition, so under unfavorable conditions, these thick walled spores will form. Okay, these thick walled spores will form. So they are thick walled and even they are non-motile. What are they, bache? Even they are non-motile. Trust me, just complete this portion related to the reproduction, then everything which is written in NCERT will be easy to understand. You just need to focus on the examples, that's all. Okay, then you just need to focus on the examples. So kindly pay attention. So chlamydospores, under unfavorable conditions they will form. They are thick walled, non-motile spores, right? These are thick walled, non-motile spores. So the diagram is going to be like this, something like this, right? So they are going to be thick walled. Okay, thick walled. Chlamydospores they are. So yes, they can ask you a question that what type of spores are chlamydospores, right? Thick walled they are, they will form under unfavorable conditions, right bache? They will form under unfavorable condition. And yes, you can even take the example in Rhizopus, you will see it. You know na, Rhizopus, it's a very common example. It's a dung mold. It's a dung mold. It can grow over the dung. Coprophilus. Sorry, not the dung mold, bread mold. Rhizopus it is. Bread mold, right? We used to see some, outgrowths on bread na. If you will keep a piece of bread outside for some time, obviously you are going to see this. Okay? Done? Done. So the last is oidia. I think this is what I have explained. The last is what? Oidia. Right? Oidia. So bache, these are not so important, but still I'll tell you. So these are the spores which will form in sugar rich conditions. They form in sugar rich conditions, right? Whenever fungus is growing at a place where too much of sugar is there. So again the examples are Rhizopus and the mucor, the dung mold. Okay, that's all, that's it. So this is about the asexual reproduction. So let's have a quick revision. Just focus here. So we started with the, yes, we started with this part: vegetative, asexual, sexual. Vegetative, asexual, sexual. So in vegetative, fragmentation, fission, budding. Everyone just repeat it: fragmentation, fission and budding. Asexual reproduction: zoospore, your sporangiosspore, conidia, chlamydospore, oidia. Zoospore, sporangiosspore, chlamydospore, your conidia and oidia. And sexual reproduction: oospore, ascospore, basidiospore. Oospore, ascospore, basidiospore. Oospore in Phycomycetes, ascospore in Ascomycetes, basidiospore in Basidiomycetes. And when it comes to your Deuteromycetes, it is having no, right? It is having no sexual stage. The members of the class Deuteromycetes, they do not possess the sexual stages in their life cycle. That is why we call them as fungi imperfecti. Is that clear? It's a repeated question, bache. Okay, it's a repeated question. Is that clear? Sure. And let me tell you about this Basidiomycetes. It is not having any asexual reproduction, right? You will not see asexual reproduction in the case of Basidiomycetes. That's all. Fine. So now, bache, when it comes to the sexual reproduction, a very important topic it is. You should know about certain. You need to know about what? You need to know about the different steps. Okay. So see, I will share the PDF. Both the PDFs will be shared today. Okay, after the class, right after the class. And as I said, tomorrow at 4 PM, there is a special class which is scheduled on Unacademy platform. You just need to download the app and directly from Google, you have to register your number or email. You guys can be the part of this. That class is free of cost, right? Right, free of cost, you people can join that class. and that class is very important for the students of NEET Avengers or Energized batch, because we are going to discuss the questions of cell cycle and cell division there. Fine. Okay, so now kingdom fungi, as I said, unicellular, eukaryotic or eukaryotic, unicellular, multicellular. You know that unicellular is rare. Yeast is the example. Fine. They lack chlorophyll, so achlorophyllous, told you. They prefer to grow in warm and humid. Done. Rigid cell wall, yes. How can I miss this point? So it's a repeated question again, bache. So see, the cell wall is rigid, which is made up of chitin and glucans. Right, the cell wall is rigid, which is made up of chitin. So here also they are having the cell wall, but the cell wall is not made up of cellulose, it is made up of chitin. So chitin is what? Chitin is a fungal cellulose. Okay, chitin is also known as fungal cellulose. This question was even there in NEET 2023, right? And this chitin, bache, it's a homopolysaccharide. What is the meaning of homopolysaccharide? Homo means same. So polysaccharide means it's a polymer. So it is a polymer. Homo means same, poly means many, saccharide means sugar. Homo means same, poly means many, saccharide means sugar. So when you talk about the chitin, it is made up of same type of monomers or same type of sugars. Right, it is made up of same type of monomers, same type of sugar. So that monomer is like it will all the monomers will be same, and that monomer is your N-acetyl glucosamine. N-acetyl glucosamine, that is your NAG, N-A-G, right? That is your NAG, N-A-G, N-acetyl glucosamine, N-acetyl glucosamine, right? So it is a very important question. Chitin, which is a fungal cellulose, right? So even you know that chitin is present in the exoskeleton of the insects also, right? It is present in the exoskeleton of the insects also. So up to this part all clear, bache? Up to this part all clear? Yes. So just look at these structures. So aseptate and cenocytic it is, septate it is, right? So nutrition is already done, saprophytic, parasitic, symbiont. In symbiont, we have the example of lichen. Even we will talk about the mycorrhiza. Any doubt here, bache? Yes, any doubt? So lichen and mycorrhiza is the example. Clear? Lichen and mycorrhiza is the example. Done? Bachcho, kindly do not chit chat. Please focus here. I know the chapter is boring. है ना? So many names are there, ma'am. So many names are there. What will we do after knowing that, okay, fungus is having the mycelium? It is of no use for us. I know that. है ना? I know that. You must be thinking, ma'am, why are we studying this? It is of no use for us. But actually, those who used to take admission in class 11th and 12th biology, they are not always interested in becoming the doctor. Some of them might be, they are interested in research, they want to go for the taxonomy, right? They want to go for the basic sciences. So in that case, we need to know about all the things. Okay, that is why. So I know it's quite boring for you people to, remember so many names, and they are even difficult also. Sometimes we are not able to, remember that chrysophytes are in Protista or ascomycetes in fungi. It takes a lot of efforts, but we have to do it. We do not have any choice, isn't it? You have decided that you are going to give the NEET examination. So if you have decided it, so obviously just give it a try, right? Thanks, bache, thanks. And see, in the first class, we used to have 300, near about 300 students. Now in second class, we have 58 students. So I can see your interest. So please take these chapters seriously. Or else, at the end, you all will watch the videos. How to crack the NEET examination in last 3 months, in last 2 months, in last 15 days, in last 15 hours. Right? Or can we crack NEET examination in last 5 months? So what is the need? What is the need to see such videos? And even I am going to make the one video. I'll be very honest about it. Right? Mostly I avoid to make such videos that how to crack the NEET examination in last 3 months. Till 6 months I'm okay. Okay, fine, in last 6 months if you follow the proper strategy, still you can do it. Right? Sometimes in last 3 months, okay, with a heavy heart I can say yes, you can do it. But then it is not possible to get the aims if you don't know anything from your syllabus. If you know something, if you have prepared earlier, then okay, otherwise there is no chance. So I have seen the videos like this. Want to crack the NEET examination? Strategy 0 to 650. Watch this. And seriously, you guys are going to watch that, and obviously we are going to get the views. We are going to get the views, and it is all because of you. And trust me, if I will post things like that, all about aims that you need to know, all about AFMC that you need to know, okay, this is the trick for memorizing the planned kingdom example, this, I'm not going to get the views. But if I will post how to crack NEET examination in one month, how to crack NEET examination in 2 days, trust me, I will get so many views. Why? Because you all want things very easily. You don't want to work hard. You all need a shortcut, which is not possible. Trust me, it is not possible at all. Students are spending 2 years, 3 years, 4 years for getting admission in AIMS Delhi, in AFMC, in JIPMER, in CMC, whatever college it is. How can you even think that you guys can get admission right after just completing your syllabus in last 6 months? And I am telling you, today you are saying, yes ma'am, we agree. But again, if I will make any such video, you are going to watch it and you will give me the views. Right? And I literally feel bad when I used to see such strategy videos are getting so many views, and a teacher is giving his or her best, and they are teaching you from last 3 hours, 4 hours, 5 hours, 6 hours, and they are teaching you in detail. They are giving you everything possible, right? For this content, you used to pay 2 lakhs, 3 lakhs. Do you know that? Do you know that? You used to pay 2 lakhs in the offline coachings. But because of this, right, because of this YouTube, because of this technology, you guys are getting it in. I think you just have to pay for your internet, that's all, right? And we don't get views on such type of videos. You know, these days if a student want to crack the NEET examination, they can just crack it by just following the YouTube video sincerely, plus that student need to be dedicated, they need to practice the mock test. So if that student is sincere, and let's say you are sincere, you know that you have to finish the planned kingdom, okay, you are checking out my video, you are like, now I have to go for the animal kingdom video, you checked my video, you were not happy, you were like, no, I want to study from another teacher because I want to finish this chapter. You can check 3 to 4 videos. If you feel comfortable with any other teacher, you will go and check it, just because you just focus on the content. This is also a very good approach. I'm not against it. But yes, if you are checking planned kingdom taught by me and from other teacher also, then you are comparing, then you are wasting your time. Okay, so if you are that much focused that today I have to complete this chapter, that chapter, this chapter, that chapter, okay, you want to change your teacher, it's fine, it's totally fine, right? It's totally fine, but you should have that sincerity. So instead of giving views on such videos that how to crack exam in last 6 months, 4 months, 3 months, 2 months, 1 month or 15 days or even 2 days, or even how to revise for the NEET examination in last 8 hours, then obviously it's not good. It's not good. Instead of that, start watching such videos where teacher is actually teaching you. Okay, literally. This batch, if any student follow us sincerely, because we are not going to drop this batch, if you guys are going to follow us sincerely along with question practice, along with NCERT reading, obviously you will get good marks. In biology, I can say whatever I am teaching you, providing you, obviously it is too much for getting 360 out of 360. But again, it depends upon you. I always used to say, okay, teacher can just guide you, teacher can just tell you this is what you need to study, and or I can make a topic easy for you, I can provide you the notes, the good notes, that's all. But ultimately it is the student who's going to put the efforts. You have to revise it, you have to read the NCERT, you have to solve the questions. You are the game changer. Okay, if any teacher is telling you that okay, if you will follow me then only you will crack it, no, it's always the student's effort. Always you guys are going to do the magic. Okay, you people can do it, trust me, but be sincere. Okay, be sincere. Be obedient. Yeah, don't watch such videos. I am telling you, don't watch such videos. Being a teacher, who's going to make such videos? Is telling you, please don't watch such videos. I'll be happy when I'll say, when I'll see that my academic videos are getting very good views and my strategy based videos are getting very low views. I'll be very happy. I'll be very happy because okay, now students know what they have to watch. Fine. So sudhar jao, sudhar jao. Now come back. So we are talking about the symbionts, where two organisms are living in a symbiotic relationship where both are getting the benefit from each other. It is basically the mutualism as well. So in the case of fungi, these are the two examples. Okay, these are the two examples, lichen and mycorrhiza. Lichen and mycorrhiza. Clear bache? Clear bache? Done? So sorry. Okay, so so here you can see when it is asexual reproduction, ultimately sorry, spore formation is there. Now when it is the sexual reproduction, what we have to do, see, there are 3 steps. Okay, there are 3 steps. Basically, first of all, ultimately it is the mycelium that we need. Let's say this is one mycelium, let's say this is another mycelium. Maybe both are having the same strains, and even it is also possible that they are having the different strain. Okay, it is also the possibility. I'm not going to tell you this thing in depth. So very first thing is fusion. Fusion here. Like let's say, just take one example, because you will see this na, ascomycetes. Okay, first of all understand this. Now there is the plasmogamy. The very first step of sexual reproduction is what? It is the plasmogamy. Gamy means fusion. Plasmogamy. Let's say plasma, protoplasm is the word. Remember it in this way. Okay, I'm just telling you this trick so that you will be able to remember it. So now you are fusing it together. This will be the case. Basically you are fusing the cytoplasm together. Yes, that will be better. You are fusing the cytoplasm together. Okay, this will be the case, yes or no, bache? This will be the case, yes or no? Yes or no? Now, there are 2 nuclei. There are 2 nuclei, isn't it? There are 2 nuclei. So, can I not say that it's a dikaryon stage? Di means 2. Understand the meaning. Di means 2. Karyon means nucleus. Di means 2. Karyon means nucleus. There are 2 nuclei. There are 2 nuclei. Yes or no? So the very first step is your hyphae, right? Their cytoplasm will fuse. Okay. Now each this compartment is having 2 nuclei. So it will be dikaryon stage. What is it? It is dikaryon stage. 2 nuclei are there. Bache, I already told you that thallus, right? Thallus of fungi is haploid. It is going to be haploid, that is N. So if I have 2 nuclei here, so it will be N plus N. It cannot be N multiply N. It is N plus N. What is it? It is N plus N. So this is what? This is dikaryon stage. 2 nuclei are present here. 2 nuclei are present here. Are you getting it? 2 nuclei are present here. Dikaryon. Now in some fungi, this stage can last for a longer period. And overall, we used to call it as dikaryophase. Like first phase is your, first thing is your plasmogamy. After plasmogamy, there will be the dikaryophase. Dikaryo, 2 nuclei. So basically in the mycelium, you will see 2 nuclei, and that is N plus N. So even if you are saying dikaryophase, dikaryon stage, it is one and the same thing. Now in some fungi, it will remain like it for some time, and after that what is going to happen? The fusion. After then what will happen? The fusion. Now the next step is karyogamy. So again, gamy means fusion, karyo means nucleus. Gamy means fusion, karyo means nucleus. Again, what is the next step here? Karyo means nucleus, gamy means fusion. So there will be the fusion of nuclei, right? Now it is N plus N. So obviously when it will fuse, it will become 2n, yes or no? Then it will become 2n. In dikaryon phase it was n plus n, but now it will fuse, it will become 2n, yes or no? It will become 2n. Now after that, this diploid cell, it will undergo meiosis and we will be having the further spores. So this is just an overview, bache. See here, as such from NCERT, right? So the very first thing, plasmogamy, there will be the fusion of protoplasm of two motile or non-motile gametes, right? It can be like this, or hyphae can get fused, right? This is plasmogamy. It is the first step of sexual reproduction. After plasmogamy, there will be the karyogamy, right? But in between, there will be the n plus n phase. That is your dikaryon phase. That is your dikaryon phase. So after plasmogamy, nuclei of both gametes, they will fuse together, karyogamy. So after karyogamy, it will be diplophase. Diplophase means now the nucleus is diploid, right? Nucleus is diploid. So last step of sexual reproduction involves meiotic division in zygote. So here also zygote will undergo meiosis. So can I not say that it is the zygotic meiosis? Can I not say that it is the zygotic meiosis? Yes or no? Priya, tell me, can I not say that it is the zygotic meiosis? Of course it is. So haploid spores will form, that spores will again germinate. Again you will get the thallus, right? Again you will get the hyphae. So basically these are the three steps in the case of sexual reproduction in fungi. It is written here, dikaryophase is a condition of having dikaryon in an intervening dikaryonic stage. I think I have already explained it. So again bache, again I am drawing it. Let's say two gametes are there, or even you can say two hyphae are there, right? So that hyphae can have same strain, homothallic it will be. If the different strain is there, heterothallic it will be. If the different strain is there, heterothallic it can be. So now let's say these two, let's take the example of gametes. So as I said, in first stage, plasmogamy. So n plus n, n plus n, dikaryon stage. Then as I said, in some fungus it will stay for a longer period, and then there will be the karyogamy, nuclear fusion. So now this n plus n will become 2n. Then it's a kind of obviously zygote. So zygotic meiosis will be there, or you can simply mention the word meiosis. So you will get what? Haploid spores. Haploid spores. And these haploid spores will do what? Germination. I think now it's easy. Germination. Is that clear? Yes, is that clear? So this is it. This is the sexual reproduction in the case of fungi. If there is any doubt, please let me know. Yes, bache, please let me know. Yes, bache, clear? Sure, sure. So bache, now we do not study that things in detail, and even it is not given in your NCERT. But when you talk about this plasmogamy, na, right, if in detail, if I tell you about these plasmogamy, it is also having different ways, like very easy ways, right? I'll focus here. I'll just give you the overview, pakka. So when you talk about the plasmogamy, here also different ways are there. Out of that, simplest is for fusion of gametes, out of which one gamete is what? It is having flagella, it is motile, right? Let's say one gamete is what? It is motile. So it is the simplest type of plasmogamy. What is it? It is the simplest type of plasmogamy, and both the gametes obviously opposite they are. They belong to the opposite strains. Okay, both the gametes here, right? So basically they belong to opposite strains. So this is also a simplest way of doing this plasmogamy. Okay, the opposite strain is here. So obviously after it, what will form? The diploid zygote, right? What will form? The diploid zygote, the simplest way. right? Now one way is gametangial contact. Do you know that? Gametangial contact. Are you aware of it? Ultimately the first step is going to be same, the plasmogamy. But sometimes you know some different structures are formed. Just gametangia is a structure in which gametes will form. Right, bache? When it is the gametangial contact, so what is going to happen? Two gametangia, they will contact to each other. Right, two gametangia, they will contact to each other. Gametangia is a structure in which gametes will form. Right, they will contact to each other. Gametes will form. So here you can just take this example. So here what will happen? The fertilization tube will form. So if fertilization tube will form, right, a small fertilization tube will form that will help in that connection. So obviously, so when there is the small fertilization tube, here also you are having the nuclei, here also you are having the nuclei. Again the structure will fuse, plasmogamy will occur, N plus N phase will come, right? N plus N phase will come. So such example is there in the albugo. Yes, albugo. Clear, bache? So there are different ways of this. Okay? Okay? Done, bache? Done? So we are not going to discuss that in detail. So here with the help of fertilization tube, the content will pass here. Sometimes this is gametangial copulation where both of them will get fused. So different ways are there for that plasmogamy. So they are not discussed in NCERT, so I am not teaching you this part in detail. So I am just telling you that there are 3 steps for the reproduction in fungi, and that is what you need to remember. Okay, there are 3 steps in the reproduction of fungi. That is what you need to remember. Clear, bache? Done? Done? Yes? Okay. Done. So phycomycetes, common name is algal fungi. Now bache, in this phycomycetes, I told you before that in the case of NCERT it is not divided, but actually it is having 2 things, oomycetes and zoomycetes, right? So basically it is divided. It is divided into 2, oomycetes and zygomycetes, oomycetes and zygomycetes. Okay, and this oomycetes is majorly, it is called the algal fungi, right? It is called the algal fungi, and let me tell you, oomycetes, quite primitive it is. So here we used to have cellulosic cell wall. This is, you can also note it down like an exception, right? In the oomycetes we used to have the cellulosic cell wall, right? In oomycetes we used to have the cellulosic cell wall. So you guys can even note it down like an exception. Okay, like an exception. Clear, bache? Clear? Okay, so cellulosic cell wall is there, and when it is the zygomycetes, again everything is same. So ultimately here in oomycetes you have the example of albugo, you have the example of phythium, right? They both are disease causing, right? They both are disease causing. So I am just quoting you one example. In zygomycetes you have the example of rhizopus, your bread mold, mucor, your dung mold. These are the examples. So are you getting it? So because it is not explained in detail in NCERT, so that is why I am not touching it, but at least you people should know what is the case here. So 2 words are there, oo and zygo, oo and zygo, and it is zygomycetes. So obviously here, okay, that I will write on another slide. Is that clear? Is that clear? So overall, if I talk about the points from NCERT, it is simply mentioning ki algal fungi, obviously they both will be aseptate and cenocytic. I told you already. Asexual reproduction can be by a zoospore, it can be by a aplanospore, it can be by a chlamydospore, it can be by a sporangiospore. So basically different members are there, they can opt different ways. I told you sexual reproduction will be isogamy or oogamy. Iso means bache same, gametes are same, right? What is the meaning of isogamy? Isogamy means, iso means same type of gametes are there, size, shape, everything is same. Physiologically they may be similar or dissimilar. Morphologically they are same. Physiologically they may be similar, may be dissimilar. And when it is oogamy, it is sorted, right? Your male gamete will be motile and small in size. Female gamete will be large and it will be non-motile. It is large and it is non-motile. That's the point. Clear, bache? That's the point. Clear? Clear, clear, clear? Is that clear? And fruiting body will produce zygospore. These are the examples. So this is as per NCERT. Am I right or wrong? This is as per NCERT. Am I right or wrong? Now, let me add extra points. Right? I will only add the required extra points. Right, bache? So don't you worry about it. Don't you people worry about it. We are going to relate it with directly with the NCERT. So you guys can see here. Okay. You people can see here. Fungi. It's a unique kingdom of heterotrophic organisms. Yes, ma'am, we know that. Great diversity is there. Yes, we have already discussed. Right, mushroom, toadstools, they are also the fungi. Yes, white spots seen on mustard leaves are due to a parasitic fungus. White spots. Right, do you know that? White spots. Albugo. It is the albugo. Yes or no? Bache, this is your homework. This is your homework. You have to tell me the name of that parasitic fungi. And bacho, I want to see many comments. Okay. You are not doing your homework. So you have to name this parasitic enzyme, parasitic fungi which is causing the white spots here. Right. So unicellular is also done. So see, Puccinia is an important example. We will discuss it. Antibiotics we are getting from the fungus, penicillium. They are cosmopolitan. Cosmopolitan means present everywhere. They are present everywhere. Right, bache? So they prefer to grow in warm and humid places. So have you ever wondered why we keep food in the refrigerator? To prevent food from going bad due to bacterial fungal infections. We have already discussed it. Right, bache? So when it comes to the body, hyphae is there. Right, that is forming the mycelium. We have already completed this part. And these 3 steps also. That fusion of protoplasm is plasmogamy. 2 nuclei when they will fuse, karyogamy, and then there will be the meiosis. Clear? Clear, bache? So, these are few examples like mucor, Aspergillus agaricus. So, mucor, it is from Phycomycetes, this Aspergillus from Ascomycetes, this agaricus from your Basidiomycetes. Clear, bache? From your Basidiomycetes. Is that clear? Now come to, where is Phycomycetes? Don't tell me that Phycomycetes is not there as per NCERT. Okay, so bache, might be it is not there, but theek hai, okay. But let me discuss the points here. Might be it is not here, koi baat nahi. So next is what? Phycomycetes. Right? Quick points. So Oomycetes and Zygomycetes. Oomycetes will have albugo. The example is given in your book. Here you have Rhizopus and mucor. Okay. So here asexual spore. Only the important point, bache, I am telling you. Zoospore. Here the sexual spore is oospore. Cell wall is cellulosic. Right, cell wall here is cellulosic. Is that clear? Cell wall here is cellulosic. Is that clear, bache? Yes, everyone. Albugo. Clear? Clear? And obviously both will have aseptate or cenocytic hyphae. Right? Cenocytic hyphae. Done, bache? Is that clear? So this albugo used to cause, it is important, white rust of crucifers. It is responsible for causing this disease. White rust of crucifers. It is important. Okay, it is important. So it used to cause white rust of crucifers. Now here we have the zygomycetes, right? Here we have the zygomycetes, rhizopus and mucor is there. So asexual spore is your sporangiospore, right? It is your sporangiospores. In bread mold we used to see this. Sexual spore. Sexual spore is zygospore. Chitinous cell wall is here. Both are cenocytic. Clear bache? Both are cenocytic. So these are the points that you need to remember. Even examples are also given. So if there is any doubt, do let me know, bacho. If there is any doubt, please let me know. Clear, clear, clear. Yes, bache, is that clear? Yes, bache. Is that clear? Sure? Sure, bache? So this is about the phycomycetes. The points for the quick revision. Now obviously what do we have? We have the ascomycetes. Next is what? Ascomycetes. Sac fungi. Right, what is it? It is the sac fungi. Now in the case of ascomycetes, you even have the unicellular fungus, that is yeast. Okay, you have the unicellular fungus, that is yeast. So bache, see, I told you now that let me explain you the asexual and sexual reproduction, and then the chapter will be very easy for you. Now see, this is how we have to make the notes. Right, this is how we have to make the notes. So there will be the quick revision. If you know the meaning of this, sexual, asexual spore, which spore is there, which spore is not there, the steps, obviously it is going to be very easy for you then. Right, it is going to be very easy for you then. Okay, so now is what? Now is ascomycetes, so you know that it will be aseptate. It will be aseptate. There will be the simple pore in between. Yes or no? It will be the aseptate. There will be the simple pore. Now when you talk about the asexual spores, right, now when you talk about the asexual spores, so conidia is very common. Right, conidia, you remember that branches. Do you remember this, bache? Conidia, exogenously. Right? Conidia, C for chain. So, exogenous spore it forms exogenously. So, it is very common. Right? You will see conidia in the case of aspergillus. Right? There is one example, aspergillus. It is given in your NCERT. Your penicillium. Right, bache? Penicillium. In their case, you will see this. And if you talk about the yeast, in the case of yeast, budding will be there. Right? Budding in the case of yeast will be there. Fine, bache? Budding in the case of yeast will be there. Okay? So now, okay, this branch is what? It is conidiophore. I think I have already explained you. There is no need to go in the depth of that. So now what you need to include in your NCERT or in your notes, that when it comes to the aspergillus, from aspergillus, right, it is also known as the weed of lab, right, weed of lab. We have neurospora also, it also belongs to the ascomycetes. There is one more example of ascomycetes, neurospora, which is known as Drosophila of plant kingdom. Note down, everyone just note down. Neurospora, which is known as Drosophila of plant kingdom, it belongs to ascomycetes. Why Drosophila of plant kingdom? Because many experiments related to, you know, this chemical things, they are conducted on the neurospora. And this aspergillus is the weed of lab. And moreover, from this aspergillus, citric acid. We used to get citric acid. Okay. This is important. Please note down. It is there in 12th syllabus. Right? So, because it is used in Srinath, many experimentation, that is why. So, now when it is penicillium, so penicillium, you know that first antibiotic was obtained. First antibiotic was obtained from it. Right? Penicillin. It is penicillin. Got it? And when it comes to the yeast, you know that in brewing and baking industry, right? In baking and brewing industry. But this is important, please note down. I'll tell you other points also, but as of now just note down. Just allow me a minute. Just a minute, बच्चे. Done? So up to this part all clear? These are the important things. Up to this part all clear? Now next here is that when it comes to the sexual reproduction, okay, the 3 steps are same. The 3 steps are same in the case of this Ascomycetes, that there will be the plasmogamy, there will be the karyogamy, and then there will be the meiosis. And where will this meiosis occur? In the zygote. So this is what you need to remind yourself again and again. I told you na, zygotic meiosis and the gametic meiosis is important to understand, right? That is why in Protista I explained it. So I hope you all can link it there. Okay, so now here the another important point is that karyogamy is delayed after plasmogamy. Okay, it will not take place immediately. Karyogamy is delayed after plasmogamy. So it means that dikaryon phase will stay for some time, isn't it? It means that dikaryon phase will stay for some time, yes or no? If karyogamy is delayed after plasmogamy, matlab that dikaryon phase will stay for some time, isn't it? Isn't it? And after that, after karyogamy, you know that zygote will form. After karyogamy, you know that zygote will form. Now, bache, this zygote, you know that the structure in which all that things are taking place, right? You know, in the case of Ascomycetes and Basidiomycetes, we will talk about the fruiting bodies. So here what is going to happen, right? This dikaryotic cell, that zygote, right, bache? Right, bache? So what is going to happen here? What is going to happen here, right? So basically that dikaryon, it will get converted, right? Not that dikaryon, that diploid zygote. Wait. This is Ascomycetes, na? Okay, just wait. So karyogamy is delayed after plasmogamy. This is the point. So basically in the hyphae, this will be the scenario, yes or no? You tell me about it. In the hyphae, this will be the scenario, yes or no? Tell me. Tell me. Tell me. Quick. Yes, bache? Quick. This is the thing, na? Karyogamy is delayed after plasmogamy, means this will be the scenario, means we will be having a dikaryon, right? So now what is going to happen here? This dikaryon, these cells having dikaryons, they will get converted. Like now we will use the word ascus, mother cell, for it, right? See, this is how you have to remember. I'm not saying that you have to remember the depth. It is Ascomycetes, sac fungi. In the case of Ascomycetes, you will see some this kind of structures, some this kind of structures. We used to call it as ascus. Like let's say, or wait, right? See, let me explain you this. So basically what is happening in the case of ascomycetes, you will see some structures like this, some sac-like structures, some pouch-like structures. Are you getting it? Some sac-like structures, some pouch-like structures. We used to call it as ascus. We used to call these as ascus. What is it? Ascus. Now all that ascus together, they form the ascocarp. Right? They form the ascocarp. Endogenously means basically in these ascus the spore will form. Right? Basically in these ascus the spore will form. So these dikaryon, they will, the cells having these dikaryon, they will get converted into ascus mother cell. So if it is the mother cell, I always told you that there will be the diploid cell. Now obviously karyogamy will occur. So it is the diploid cell. So it will undergo meiosis, and then there will be the formation of these haploid spores. So ultimately in this ascus, what will you see? You are going to see some haploid spores. What will you see in these ascus? You will see some haploid spore. Are you getting it? What will you see there? Haploid spores. Is that clear? So what you have to remember from this part, that in the case of ascomycetes, ascus, some sac-like structure will form. The plural is SI, ASCI, right? And here you will be having the haploid spores. Right? Here what will you have? You will be having the haploid spores. What will you have here, bache? You will be having the haploid spores. So 4 to 8 haploid spores will be there in each ascus. 4 to 8 haploid spore will be there. Okay? Right? Right? So they may occur freely, or they can form this fruiting body as ascocarp. So ascocarp is nothing, it is the fruiting body. What is the meaning of fruiting body? Right? Where these spores are forming. Right, where these spores are forming all together. So in each ascus 4 to 8 nuclei can be, 4 to 8 spores can be there, and obviously these are meiospores formed after the meiosis. Okay, these are the meiospores formed after the meiosis. Is that clear? Is that clear? This is about the ascus. Right, this is about the ascus. So see here, just let's finish it from NCERT itself. So commonly known as sac fungi, they are mostly multicellular, right? Rarely unicellular. Yeast is there, Saccharomyces. They are saprophytic, they can be decomposer, parasitic or coprophilous. Coprophilous means it will grow on dung. Now mycelium is branched and septate, right? It is branched and septate. This is how you can draw it, although it's a very bad diagram, but okay, still, right? So these arms are conidiophores, where your conidia will form. So conidia is what? It germinated with, it is the asexual spore. What is it, bache? Asexual spore. It will form exogenously. How will it form? It will form exogenously. Write down, everyone. In the chat section, all of you just write down the name of the haploid spores quick. Just write down the name of the haploid spores quick. So conidia is asexual spore which will form exogenously, and this ascospore, right? It is the sexual spore which will form endogenously. Why endo? Because it is forming inside a structure. What is the name of that structure? Ascus it is. It is ascus. Is that clear? It is ascus. Okay. So single ascus, plural is SI, and when they are arranged in different types of fruiting body, it is ascocarp. So up to this point, if there is any doubt, do let me know. Up to this point, if there is any doubt, do let me know, bacho. Please tell me if there is any doubt here. And now see, aspergillus, I told you, weed of the lab, you are going to get citric acid from it. Right? You are going to get citric acid from it. So claviceps. Claviceps is known as ergot rye. But do you know about LSD? LSD, lysergic acid diethylamide, if I'm not wrong. It's a hallucinogen. It's a hallucinogen. Right? It causes hallucinations. LSD, ergot rye, claviceps. Then neurospora, Drosophila of plant kingdom. Yes, it is known as like this. Drosophila of not the fungus kingdom, of plant kingdom it is. because many genetic based experiment will be conducted on it. Okay, right? It is used in biochemical and genetic work. And another members are your morels and truffles. They are edible. They are very healthy, very nutritious. Edible means eatable, right? They are the delicacies. Is that clear? So morels, right? Morchella it is. Morchella it is. I hope you know about it. So morels and truffles, they are delicacies. They are edibles. Like in the case of morels, we have the examples of morchella. You might have heard about it. In Hindi, we used to call it as Gucci. It's not Gucci. It's not Gucci the brand. It's Gucci. Gucci, right? In the areas where the temperature is low, right? Over the mountains, you are going to find it. Morels, Gucci, right? So when you look at it, you will be like, yeah, what is it? Their structure is like that, right? I have seen it. I have seen it in Mandi, right? District Mandi, Himachal Pradesh. So I have seen it there. So morels, Gucci. Edible it is, highly nutritious it is. So morels and truffles, what are they? They are delicacies, they are edible. So these are the examples that you need to remember, bache. That is your job. You have to remember it, okay? So claviceps, I told you it is ergot of rye, okay? So we will get one chemical from it. Yes, that is your, right? Done, bache? LSD. Okay, done बच्चे. Done बच्चे. Sure. So this ergot of rye, this example you need to check. So this ergot of rye, actually rye, you know na, rye is a crop, basically rye. So if let's say if this claviceps, if it affects that particular crop, what is going to happen? It will produce some chemicals, right? It is going to produce some chemicals in that. Let's say someone unknowingly, right? If someone is eating that particular rye, right? So what is going to happen? Even abortion can occur. That chemical is that dangerous. Okay, that chemical is that dangerous. If unknowingly, let's say any pregnant female is taking it, so even abortion can occur. So fungus infection is very bad, and you know there are many fungus. There is one fungus. You have to check the name again. It's something important, right? Please note down. It is your homework. There is one fungus which can even stop the activity of your RNA polymerase, right? It can stop the activity of the RNA polymerase. And let me tell you, even aspergillus is having many poisonous, right? It is also having poisonous species also. So that is your homework, you need to find it out, right? Okay, clear bache? Done. So next is the first of all Basidiomycetes, and then there is the Deuteromycetes. So let's talk about the Basidiomycetes, and here you people can see the examples. The escocarp. Okay. No, this example is not here. I'll show you. Bache, these pics are not proper. This is basically the Ascomycetes. It is basically the Ascomycetes. Okay, this is Morchella Gucci. This is Morchella Gucci, and this is Penicillium. Okay, this is Penicillium. This is basically the examples of Ascomycetes, and here we have the examples of Basidiomycetes. Very good decomposers they are. Right, here we have the example of Basidiomycetes. Clear, bache? Clear. Okay, so now let's talk about the Basidiomycetes. So it is club fungi. It is septate branched. Secondary mycelium is dikaryotic. Right, primary mycelium, main structure, then obviously the reproductive phase will be there. Secondary mycelium is going to be dikaryotic. You know the logic behind. Asexual reproduction is by oidea. Sexual reproduction is by basidiospores. Right. So now if you are thinking that, well, ma'am, what is this sterigmata? So obviously it is a way of plasmogamy. Right. It is a way of plasmogamy. So right now what you need to focus? You need to focus on Basidiomycetes, the most advanced just a minute बच्चे, right? So what is it? It is the most advanced fungi. What is it? It is the most advanced fungi. Are you getting my point? Right? It is the most advanced fungi and very good decomposers there. And I told you already that when it is the Basidiomycetes, in the case of Basidiomycetes, right? So the spore will be like this. The dolipore septum will be there. Around the pore you will see the bulging. So there will be the dolipore septum. Is that clear? There will be the dolipore septum in the case of Basidiomycetes. Is that clear, bache? Yes, it is the club fungi, right? It is the club fungi. Okay. Done. Fine. Okay, so basically, done. So now in the case of Basidiomycetes, you will not see any motile structure. No motile structure is present. Fine, there will be no motile structure. So obviously there is no chance of zoospore. Okay, there is no chance of zoospore when you talk about the Basidiomycetes. Fine, when you talk about the Basidiomycetes. So now let's revise it from NCERT because sexual reproduction stages are same. We just need to focus on the examples, bache. So here you people can see, right? So mainly it will be having the mushrooms. Do you know the mushrooms now? That cap-like structures, we will talk about it, right? So basically what do we have here? Mushrooms, right? Mushrooms. So bracket fungi or puff balls. So commonly known forms of Basidiomycetes are mushroom, bracket fungi or the puff balls. They are like this, something like this, right? So they grow in soil, on logs and tree stumps and in living plant bodies, right bache? So they are as parasites also. So we have the example of Puccinia and Astrilegos, so rust and smuts. They belong to this Basidiomycetes. It is important. So these are the parasitic fungi, right? These are the parasitic fungi, rusts and smuts. Rusts and smuts. What are they? They are the parasitic fungi. So in the case of rust, we have the example of penicillium, and in the case of smut, it is Astilego. Right? These two examples are important. So they do not have these dolipore septum, and actually this dolipore septum is like this. Okay, the pores, the ends of pores are swollen. So I was drawing it in a different way, like this. Yes. Okay, like this. Swollen endings are there. Okay, bache. So mycelium is branched and septate, bache. Asexual spores are generally not found. Okay, generally they are not there, but vegetative reproduction by fragmentation is very common. Okay, usually we do not see the asexual reproduction in the case of Basidiomycetes. Initially also I explained you this part. Right. So not even with the oidia, very rarely you will see. So generally it is not there, but fragmentation can occur. No proper sex organs are there, bache, they are absent, but plasmogamy is brought about by fusion of two somatic cells, right, of different strains or two vegetative cells. Okay, so definitely in the case of sexual reproduction, the plasmogamy and other stages will also be there. But here what is going to happen? Right, see, plasmogamy is brought about by the fusion of two vegetative or somatic cells of different strains of or genotypes. Okay, so resultant structure will be dikaryotic, which ultimately give rise to the basidium. So here we have the basidium. Like last time in Ascomycetes, we are talking about the ascus. Here what do we have? We have the basidium. So basidium is the fruiting body. But basically basidium, so many basidia will form basidiocarp. That will be the fruiting body. Okay, so question can come on this fruiting body. So karyogamy and meiosis will take place in this basidium, right? So it will form basidiospores. So they are exogenous, right? So let me show you the structure here. Something like this. Okay, like this. So this is the basidium and these spores are exogenous. Exogenously they will form, right? How will they form? Exogenously they will form. Clear, bache? exogenously they will form. Is that clear? Yes, is that clear? So how will they form? They will exogenously form. So basidia, they are arranged in fruiting body, that is the basidiocarp again. So agaricus, that is your mushroom. Astilego again will cause smut, Puccinia will cause rust. So these are the parasitic fungi. Is that clear? So bache, examples are important, these important features are important. So what you need to remember from basidiomycetes? Club fungi, first thing. Second thing, they do not have the asexual stages generally. Number 3, basidium will be there, right, that will form basidiocarp, and the sexual spores will form exogenously, right, the sexual spores will form exogenously there. Is that clear? Is that clear? The most advanced fungi it is, right, the most advanced fungi it is. Okay, okay, done, done, done. Fine, this is what you need to remember. So Astilego example, okay, so Puccinia, mushrooms, you know that, toadstools you need to know about. Okay, that's all, that's all. So other are the example that you need to remember, bache. So please check that example, that is important. So let me tell you about one example, Amanita phalloides. Yes, Amanita phalloides. It is also known as death cap. Okay, death cap. Their structure is like this, na, group of, there will be a cap like structure, then you know that, right? I think you all have seen the mushroom. So here you will be having that network of mycelium, isn't it? Here you will be having the network of mycelium. So this is important, Amanita phalloides. So this is your homework. You people need to tell me what this Amanita phalloides can do. Death cap cup it is basically. Okay, this is your homework. Examples are headache, but we have to remember we don't have any other choice. Literally we don't have any other choice. I know it's very difficult to remember these examples, you know, literally I agree, but we do not have any choice. So it's okay na. Let's do one thing, bache. Let's do one thing, just do one thing. Take it as a challenge. So I'm giving you this challenge that you have to remember the examples of examples of fungi only. I'm not asking for the examples of Monera, of Protista, just the examples of fungi, right? So by tomorrow, EOD, that is end of the day, you have to remember it. Do you accept this challenge? I'll check the comment section. So you have to mention it, yes ma'am, examples done. Hashtag examples done. Hashtag examples done. Tell me. So, and it should not be a formality that okay ma'am is challenging us, so let's do it. No, it should not be like this. You have to remember it in a way that you will never ever forget, right, that you never forget it. Okay, okay, sure, done. So last is deuteromycetes. Please ignore these pictures. These are from Ascomycetes. Okay. So deuteromycetes, fungi imperfecti, because there is no sexual stage of this particular fungus. And you know what? Basically only asexual stages are present here. Now let's say this is a fungus A. Let's say this is a fungus A. Okay. So this fungus A is only having asexual stages. So obviously we are going to keep it in deuteromycetes. Are you getting my point? We are going to keep it in deuteromycetes. Yes or no? We have the fungus A. What do we have? We have the fungus A. Okay. So fungus A is only having the asexual stages and we are putting it in the deuteromycetes. Okay, because it doesn't have the sexual stages. Now let's say in future I discovered the sexual stages, right? I discovered the sexual stages. So what will I do? What will I do with this particular fungus? I'll be like, go to the different class. I'm not going to keep it. So deuteromycetes is sorted. If you don't have the sexual stages, then come to me, otherwise leave it. If you want sexual stages, just leave my class. This is the scenario here. Got it? So let's say if this A fungus is just having asexual stages and I am putting it in deuteromycetes, right? In future we discovered the sexual stages, so we will throw it out from this particular class. Let's say that sexual stages are more like ascomycetes, then I'll be like, go to ascomycetes, you are not allowed here, right? You are not allowed here. This is the case, right? That is why we used to say it is the fungi imperfecti. Okay, that is why we used to say it is the fungi imperfecti. Clear, bache? Is that clear? Yes? Any doubt here? Yes, everyone. Do you have any doubt? I think there should not be, hai na? Now, the next point is, now, even it is also the possibility that let's say we are we are putting one example of fungi in the deuteromycetes. The asexual stage there in the deuteromycetes might be another fungus which is somewhere in basidiomycetes or in ascomycetes or in phycomycetes. Maybe the sexual stages we have placed here. Even it is also the possibility. So with the time, you know, more we will study, then we will get to know about it. So this is all that you need to know about this deuteromycetes, that there is no sexual stages, only asexual stages are there. Okay, only asexual stages are there, and here you guys can see. Okay, so only vegetative phase of these fungi are known. When the sexual forms of these fungi were discovered, they were moved into classes they rightly belong to. Okay, so it is also possible that asexual and vegetative stages have been given one name, like it is placed here in deuteromycetes, and another one is placed in some another class. Okay, so later when the linkages were established, the fungi will correctly identified and moved out of deuteromycetes. This is the point here. Okay, this is the point here. So once perfect stage, like sexual stage of members of deuteromycetes were discovered, they were moved to the respective class to which they belongs to. Okay, to which they belongs to. So deuteromycetes reproduce only by asexual spores known as conidia. This is what you need to remember. Right? So, bache, you have to remember the asexual and the sexual spores name. Question can be asked in the paper. Fine, bache, question can be asked in the paper. So, mycelium is again, it is septate and branched. So, if it is septate, means uninucleated. Some members are saprophytes or parasitic. Right, bache, they are decomposers also. So, alternaria. Do you know about alternaria? It used to cause early blight of potato. So, obviously, it is parasitic. Right? It used to cause early blight of potato. Now coletotrichum, it used to cause red rot of sugarcane. This is important. Red rot of sugarcane. Now trichoderma, right? Trichoderma. Remember Trichoderma polysporum. From this fungi, we extract the chemical cyclosporin, right? We extract the chemical cyclosporin as well. And even this trichoderma, it protects a plant from the root pathogens. Okay, it protects the plant from the root pathogens. Is that clear? So these are few examples. Fine, bache, these are few examples. Done? Okay. Done, bache. So this is all about the fungi. Okay, so let's talk about the symbiotic associations of fungi. So the very first name is bache lichen. So it is the symbiotic association of, what is it? It is the symbiotic association. It is the symbiotic association of algae and fungi. Okay, so both the partners will be mutually benefited. So bache, when it is the algal partner, we will use the word phycobiont, right? We will use the word phycobiont. And when it is the fungal partner, we will use the word mycobiont. So both will be benefited. Like fungi will absorb the nutrients, essential thing, will give it to algae. Algae will make the food. Okay, algae is going to make the food. So this is lichen. Okay, this is lichen, and you know that some lichens are even the pioneers, right? This starts the succession. Do you know that? Some lichens are even the pioneers. They start the succession. Like when there is a barren land, so initially lichen and mosses they grow, right? Initially the lichen and mosses they grow. So lichen, you know that exempted from 5 kingdom classification system. It is not the part of, right, it is not the part of 5 kingdom classification system. Are you getting my point? It is not the part of 5 kingdom classification system. Okay, done. So and another symbiotic association of fungi is mycorrhiza. What is it? It is mycorrhiza. So here fungal hyphae, they are associated with the roots of trees, right? They are associated with the roots of trees. Okay, so let's say these are the roots of a tree. So fungal hyphae, they are associated here. So bache, like we have the one best example, pinus tree, right? So pinus tree cannot grow, right? Pinus tree cannot grow if roots are not associated with the fungal hyphae, because pinus roots are not, right? Pinus roots are not so capable that they can, absorb all the essential nutrients from the soil. They are not capable of doing it. Are you getting it? They are not capable of doing it. So they need fungal hyphae. So fungal hyphae, obviously they will absorb the nutrient, they will give it to it, the pinus tree, and in return they are getting the shelter, they are getting the food. Is that clear? They are getting the shelter, they are getting the food. Is that clear? Okay. Okay. Yes, bache. Done? Fine, bache. Okay, so when it is the fungal hyphae plus roots of tree, the pinus tree is there na, so basically that is also the example of actomycorrhiza. Acto means bache outside, acto means outside. So these fungal hyphae, they are not penetrating the root deeply, they are not penetrating it deeply, they are just touching the outer layers of the root. Acto. Then the word is what? Actomycorrhiza, pinus tree and the fungal hyphae. Pinus tree and the glomus fungus, this is we consider. So we also have endomycorrhiza. Here we have the members of Phycomycetes. So in endomycorrhiza, the fungal hyphae, endo means inside, yes or no? Endo means inside. So these fungal hyphae, they are penetrating the root cells deeply. What are they doing? They are penetrating the root cells deeply. Are you getting it? They are penetrating the root cells deeply. Okay, endomycorrhiza. Fine, bache, endomycorrhiza. So basically these fungal hyphae will help in the absorption of phosphate from the soil, right? They will help in the absorption of phosphate from the soil. This is about the mycorrhiza. Clear, bache? So now the next is virus, viroids and lichens. So let's finish it very quickly and then we will end the session. So if there is any doubt from this part, do let me know. Yes, if you people are any doubt from this particular part, do let me know. Yes. So see, viruses, viroids, lichens, even the prions, do you know that they are not included in 5 kingdom classification system, isn't it? They are not included in 5 kingdom classification system. Yes or no? Not included in 5 kingdom classification system. Okay, so when you talk about the viruses, you know that it is a connecting link in between living and non-living organism. When we talk about the viruses, they are connecting link between living and non-living organisms, isn't it? Between living and non-living organisms. Like virus, when it is outside the body, right bache, virus is a very weird thing. When it is outside the body, right, outside the body, then it is just a protein, just an inert crystalline protein. What is it? It is just an inert crystalline protein. But when it is inside the body, it will hijack our machinery, it will hijack the host machinery, and then obviously it will divide, it will increase its number. Are you getting it? So it is going to use the host as the machinery. Are you getting it? It is going to use the host as the machinery. Is that clear? So outside the body, it is what? It is just the inner crystalline protein. It is just the inner crystalline protein. Are you getting it? Yes bachche, are you getting it? Yes or no? Are you getting it? Yes or no? So they do not have proper cell structure, not living, they are obligate. So now you know that when the word is obligate means obsessed, means strictly need their host, right? It's parasite, so it's strictly need host, right? So let's say if there is one viral strain which is dependent on the human being, it will affect the human being only. It will not survive in the body of dog. Maybe it will get mutated and then it can survive. Otherwise, it is not going to survive there. Okay, otherwise it is not going to survive there. Right, bache? So this is about the virus. So it's a very simple thing. I will tell you some very basic point basically. So virus, when it comes to its structure, bache, it can be enveloped, like outer envelope is there. It can be non-enveloped. Right, it can be enveloped, it can be non-enveloped, right? Now if I talk about the HIV virus, you might have seen the diagram. Right, you might have seen the diagram. Right, obviously it is not a proper diagram, but it's okay, fine. This time you have to manage kids. Done. So when you talk about the virus, it can be enveloped, like outer envelope can be there, or it can be non-enveloped. It can be enveloped, it can be non-enveloped. Are you getting it? Are you getting it? So now just have a look of this virus. So it is the diagram. Obviously it's a very rough diagram, but still, because I need to explain it to you. Let's say it is the diagram of HIV virus. Now it is enveloped. It is having outer lipid covering. This part that I am showing you, it is one outer lipid covering, and these points are the receptors. These things are the receptors. They will help this virus to attach to the host body and to enter there. Right, to attach to the host body and to enter there. Now when you look at the structure inside, these two are the protein coats, and we used to call it as capsid. See, these are the terminologies that I am explaining with the help of HIV virus, right? These are the terminologies that I am explaining. Now it is capsid. Capsid is what? Capsid is basically the protein coat. Capsid is what? It is basically the protein coat, protein covering. So this capsid, like let's say there is a wall. Okay, bache. Let's say there is a wall. That wall, right? That wall is having so many bricks. So ultimately this wall is made up of so many bricks. Ultimately that wall is made up of so many bricks. So individual brick is known as capsomere here. Individual brick is known as capsomere here. And that together that brick is, together that wall is basically the capsid. So what am I trying to say? All I'm saying is that when you talk about a virus, it will have the genetic material, the protein coat. Outside maybe it will be enveloped, it will not be enveloped. Anything can be possible. Right now I'm showing you the rough structure of HIV virus, that is human immunodeficiency virus. It is basically, it is you know that it used to cause the AIDS, acquired immunodeficiency syndrome. Now here when you look at it, the outer envelope is there, these are the receptors. These receptors will help this virus to get attached to the host cell, and obviously to use that as a factory to create more viruses, right? Now here we have two protein coats. It's not mandatory that all the viruses will be having two protein coats. No, it's not mandatory. But here it is having two protein coats. So for its protein coat, the term that we are using is capsid. The term that we are using is capsid. What is it? It is capsid. So this capsid is nothing, it is the protein coat made up of small subunits, and that small subunits are nothing, they are known as capsomeres. What is it? These are known as capsomeres. So these rounded capsomeres are together making the capsid. Right now, so we are drawing it like this. Just imagine, I am the genetic material. I am covered with 2 layers. That 2 layers are capsid. Right? From all the sides, I am covered with that 2 layers. That 2 layers are capsid. Are you getting it? That's the point here. Now, in this virus, the genetic material that we have is RNA. Right? So in the viruses, we will be having the genetic material. That can be DNA, that can be RNA. That can be single-stranded DNA or double-stranded DNA. That can be single-stranded RNA or double-stranded RNA. It depends. Okay, it depends. Okay, so here we are, because we are talking about HIV virus. Here we are having 2 strands. Here we are having 2 strands of single-stranded RNA. Like we are having 2, you can say that 2 units of single-stranded RNA. These 2 are single-strand RNA, right? So in HIV virus, what do we have? RNA is the genetic material, 2 units of single-stranded RNA is there, and there are other enzymes also, but here majorly we have this reverse transcriptases. Okay, so I'm writing these points. Now you know the terminology. Majority of things I have already explained here. So in viruses, when it comes to the genetic material, either it can be DNA or RNA. Do you know that, bache? In viruses, genetic material is either RNA or DNA. Either RNA or DNA. Right? Either it will be RNA or it will be DNA. And when it is RNA, it can DNA, it can be double-stranded DNA virus. It can be single-stranded. SS means single-stranded. DS means double-stranded. Same for the RNA. Right? Double-stranded RNA can be there, and mostly it is single-stranded RNA. And usually, usually plant viruses are RNA viruses. Right? Usually, I'm not saying always. Right? This is what you need to focus. Usually, plant viruses are RNA viruses. Usually, okay, and animal viruses are DNA viruses. Animal viruses are what? They are the DNA viruses. As I said, usually, not always. Now, you know about the HIV, you know about the COVID-19. Do you know about the COVID-19? Yes or no? Do you know about the COVID-19? Yes, bachcho. Remember COVID-19? Right? Do you remember COVID-19? So COVID-19 is also what? It was the RNA virus, isn't it? It was the RNA virus, isn't it? It was the RNA virus. It was affecting the human beings. So it was very difficult, you know, to make medicine against such viruses. Why? Because it is RNA virus. RNA can mutate very quickly. RNA can mutate very quickly. Are you getting it? HIV virus is also the RNA virus. So any virus having RNA as genetic material, any virus having RNA as genetic material is known as retrovirus. That is known as retrovirus. What is it? It is known as retrovirus. Okay, retrovirus. Are you getting it? So RNA viruses, they mutate very quickly, so it becomes very difficult to make medicines against it. Okay, so we have double-stranded also, single-stranded RNA viruses, double-stranded DNA viruses are also there, single-stranded DNA viruses are also there. So in the case of plants, I told you na, usually plant viruses are the RNA viruses. So there is one exception. There is one exception. Cauliflower, write down bache. Cauliflower mosaic virus, it is a DNA virus. Okay, it's a DNA virus. So this is an exception, right? So cauliflower mosaic virus, what is it? It's a DNA virus. Okay, it's a DNA virus. You can say that it is double-stranded DNA virus. Fine, it is double-stranded DNA virus. So this is an exception, and usually animal viruses are DNA viruses. Is that clear? Is that clear? Now, bache, write down one more exception. Write down one more exception. It is also a previous year question. Leucovirus. Leucovirus. It is important. It is an exception. It is an exception. It contains. See, if you read NCERT carefully, in the NCERT it is clearly mentioned either virus will have RNA or DNA. Either virus will have RNA or DNA. But leucovirus is an exception. It contains both RNA and DNA. Okay, so it is an exception. This is what you need to remember. This is what you need to remember. So what I just said, the virus having RNA as genetic material, right? The virus having RNA as genetic material, that virus will be what? Retrovirus. So in retroviruses, we used to have one enzyme that is known as reverse transcriptase. In the case of HIV, there are two reverse transcriptase enzymes, right? So what do we have? We have an enzyme which is known as reverse transcriptase. So this can form complementary DNA from RNA. It can do the reverse transcription. From RNA it can form the DNA, right? From RNA it can form the DNA. Is that clear? Right? From RNA it can form the DNA. Is that clear? Yes or no, bache? Yes or no, bache? Okay, so one more virus, this is your T2 bacteriophage virus. So bache, this is bacteriophage. The name is having bacteria in it, but actually it's a virus. So this T2 bacteriophage is a DNA virus. It's a double-stranded DNA virus. Okay, it is having this tadpole-like structure. Tadpole is the larva of frog. Okay, so here surrounding is basically the protein coat. Okay, it is the protein coat. Basically, it is a head region. Right, it is the head region. It is the genetic material. So this is given in your NCERT, and it is your homework to read it from NCERT. Clear, bache? It is your homework to read it from NCERT. Clear? Yes, bache? Is that clear? Okay, so it is having this head. It is having here neck. It is having one collar as well. So this is the tail here. So here you are having the tail fibers. So that is the structure, and you have to remember it. So it's a bacteriophage. It's a double-stranded DNA virus. It can infect bacteria. Okay, so this is a virus which can infect bacteria, right? So it's a kind of hyperparasite which is parasiting the parasite. Okay, so it is a kind of hyperparasite. And one more diagram is given in your NCERT. You know, some structure like this. And then the first virus discovered. Basically, I will tell you about the scientist names also, something like this, right? So I hope you know about it. It is your TMV, tobacco mosaic virus. So it is having single-stranded RNA. So it's a RNA virus. So these are basically the capsomeres which are making the capsids. These are basically the capsomeres which are making the capsids, right? So this is about the viruses, bache. All the general terminology I have already explained you. They are responsible for causing many disease. So you have to write 10 disease caused by the virus. Okay, you have to write 10 disease caused by the virus. Okay, okay. So if I talk about the term, you know what? Term virus was coined by, virus means venom. Venom means poisonous, right? Venom, which is poisonous. So it was discovered by Pasteur. You know that? The term was discovered by Pasteur, but properly DJ Ivonsky, he's a Russian, right? He's a Russian scientist. He discovered the virus. Okay, he is a Russian scientist. He discovered the virus. Okay, so basically, bache, there are certain filters, right? Remember during the COVID time, we used to say that we have to use the N95 mask, this mask, that mask, because that is having very small size, right? So virus cannot cross it like this. So similar way, we have some, you know, bacterial filter, virus filter. So Ivansky, he discovered the virus. Which virus he discovered? TMV, that is your tobacco mosaic virus. So what happened was, he found something and it was very small, right? It can even cross the bacterial filter. So he was confused that, okay, it is something smaller than bacteria which can even cross the bacterial filter. Its size is so small. So he recognized certain microbes causing mosaic disease of tobacco. Basically it was tobacco mosaic virus. So he found that it is smaller than bacteria and can pass through bacterial proof filters. Okay, then comes your M.W. Wejernik. These scientists' name, it is important. So he demonstrated that extract of infected plants of tobacco can cause infection in healthy plant, right? So one tobacco plant was affected, it was having the cells, so from that cells he collected some sap, so that sap was infectious. So he called the fluid as Contagium vivum fluidum, infectious living fluid. It is important to remember, bacha. It is very important to remember. Are you getting it? It is very important to remember. Are you getting it? Yes, bacho, are you getting it? So next, M.W. Stanley, he showed that viruses could be crystallized and crystal contain largely of proteins. So these are the discoveries and the name of the scientist, and we are not allowed to miss it. And here you guys can see, it is just having protein coat, right? It is having lipid envelope as well. So enveloped viruses, non-enveloped viruses, I told you already. So all that points I have already explained, that they can have different shapes, bache. Protein coat is capsid, small subunits are capsomere. Capsomere can be helical or polyhedral, right? So virus is nothing, it's nucleoprotein, your nucleic acid plus protein, and the genetic material is infectious. Is that clear? Genetic material is infectious. This is the virus. Clear, bache? Have a look of different type of viruses. Their shape also vary. So these receptor will help in entering the host cell. Okay, in HIV virus we will discuss it. Glycoproteins this, glycoprotein that. One will help in entry, one will help in, you know, attachment. We will talk about it. Clear, bache? See, this is the diagram which is given in NCERT, TMV. So it is single-stranded RNA virus. It is double-stranded DNA virus, right? It is double-stranded DNA virus. Clear, bache? And it is single-stranded RNA virus. RNA virus. Okay. So then is the viroids, bache. Viroids. How to remember this? Viroids. Viroids, it is smaller than virus and it is basically the infectious RNA or infectious genetic material. This is what you need to remember. We know the virus, we have discussed the virus. Next is viroids, prions. Viroids, prions. Viroids are infectious RNA. This is how you have to remember. Viroids, infectious RNA. Infectious, the RNA can cause the infection here. So obviously it is not having the RNA, the genetic material and protein. No, it is just having RNA. If you compare it with the virus, virus is having the genetic material covered by protein coat, and that covering is important to protect the genetic material, right? Manik, bache, are you getting it? But here what is the story? Only RNA is there. There is no protein coat, and that RNA is infectious. So they are infectious agent that are smaller than viruses, caused various diseases. One is PSTD, potato spindle tuber disease, right? One disease here is PSTD, potato spindle tuber disease. So yes, question can come in paper that which disease is caused by which infectious agent. So viroid is causing PSTD, right? Potato spindle tuber disease. And one more is there, chrysanthemum stunt. So this is it. So it is free RNA which is infectious without any protein coat. Okay, so its molecular weight is low. So next is bache, in plants I told you chrysanthemum stunt, potato spindle tuber disease. Next is prion. Now P means protein. P means protein, but here the protein is infectious. Here the protein can cause the infection, can cause the disease. So basically it is abnormally folded protein. What is it, bache? It is abnormally folded protein. What is it? It is abnormally folded protein. Are you getting it? It is abnormally folded protein. So in modern medicine, certain infectious neurological, so see, these abnormally folded disease proteins can affect our brain directly. They cause very dangerous disease. One is your, your laughter disease, Kuru disease. That is known as your Kuru disease, laughter disease it is. CG disease, right? One is your, that mad cow disease. So yes, you have to remember the name. So basically here, right? So its size is just like viruses, but they are just abnormally folded proteins. So in animals, scurvy in sheep, mad cow disease, chronic wasting disease in deer, right? This mad cow disease is also known as bovine spongiform encephalopathy, right? It is also known as bovine spongiform encephalopathy. Okay, encephalopathy. In humans, it is CJD disease, fatal familial disease, Kuru disease, also known as laughter disease. So see, these abnormally folded protein can damage our brain. Okay, they can damage our brain. Are you getting it? So yes, of course, here you have to remember the name of the disease. So lichens, I have already explained you, bache, two partners will be there, phycobiont, your algal partner, mycobiont, your fungal partner. So algal partner will cook food, fungal partner will give it the nutrition. That's all, right? That's all. So lichens are also the good pollution indicator because they do not, right? They do not grow wherever there is the pollution. So we are done with the chapter. So how are you people feeling? I know when we started the biological classification, we started with the, right? We started with approximately, I think 200 plus students were there. If I am not wrong, 200 plus students were there. Okay. Okay. So now 42. But we don't mind. We are here to teach you. So start revising properly. That's all. That's it. Now answer the question. Answer the question. Then we will end the session. Even I am hungry. Okay. I need to eat my dinner. So which of the following are found in extreme saline conditions? Very easy it is. Yes, so many things are there. You have to write down the name of the DNA viruses and RNA viruses, 5 examples each. You have to write down the 10 disease caused by viruses. You have to write down the 5 disease caused by protozoa. The challenge is you have to remember all the examples of fungi. And you have to revise it because not tomorrow, on Tuesday, I am going to take the problem practice session in which the questions will be from living word and biological classification. And tomorrow at 4 p.m. on Unacademy platform, there is one special class where we are going to revise cell cycle and cell division. And moreover, guys, if you are not the part of Avengers batch or the Energize batch, guys, join that batch. You are going to love that batch, mark my words. You people are going to love that batch with the same energy, same enthusiasm. But yes, in that batch, students mostly, they, whenever they ask stupid questions, I even used to scold them. Obviously, wherever there is a love, concern, anger, everything is there. Okay, so be the part of that batch, but don't be too late. Otherwise, there will be, backlogs. So start studying properly. Arey, no, good night. Answer the question. Which of the following are found in extreme saline conditions? What should be the answer here? Guys, answer the question, then we will say bye-bye, tada. Of course, it is archaebacteria. So here we have the next question. Single-celled eukaryotes are included in. Single-celled eukaryotes are included in. Single-celled eukaryotes are included in. Again, it is A, that is Protista. Next question. Chrysophyte, euglenoids, dinoflagellates and slime molds, they are included in the kingdom. Again, it is A, Protista. Next. A few Protista possess structure to regulate their water content. They are, okay. A few Protists, they possess structures to regulate their water content. Salt and water content basically. So they are contractile vacuoles. They are contractile vacuoles. Very good. So the next question, pick up the wrong statement, bache. Just pick up the wrong statement, all of you. So diatoms are primarily produced in oceans. They are microscopic and float passively in the water. Walls of diatoms are destructible easily. Diatomaceous earth is formed by the cell wall of the diatom. So what should be the correct answer? What should be the correct answer? Guys, be quick. What should be the correct answer? Oh, some students are saying D. It's not D. It's C. Walls of diatoms are not destructible. They are not destructible. Okay, next, the fungi division includes club fungi. So club fungi is basically yes, basidiomycetes. Next question, what does perfect stage of the fungus indicates? Oh, indicate. What does perfect stage of a fungus indicate? What does the perfect stage of a fungus indicate? Of course, it indicates that it is having the sexual stages. Fine. So the group is used to represent pathological fungi. Tell me, which group is representing the pathological fungi? Pathological, which can cause the disease. Yes, bache, which can cause the disease? Again, it is C, smut, rust and molds. So thank you so much. Thank you so much for your time, bacho. So I will share the PDF in my Telegram group. Okay. So now you can use this PDF for your quick revision. Okay, so you have to join the class. I will, it is scheduled for Monday 9 PM, but Shreyas sir is having a lecture. So I will schedule it for Tuesday and you will get time for the quick revision of this particular chapter as well. Okay, so just be sincere and take care of yourself, be regular and please utilize, right? Utilize this content. Please start revising properly. Okay, please start revising properly. And I want to see these messages in the comment section now and all your homework in the comment section. Fine. So take care. Thank you.